Ahí donde está el estrado.
¿Listo? Hola, muy buenos días, estimada comunidad del IBT y quienes nos acompañen. Es un verdadero gusto iniciar esta serie de simposios de los 5 departamentos del IBT como parte de los festejos del 40 aniversario de nuestro instituto. Hoy el Departamento de Microbiología Molecular ha generado un extraordinario programa que podemos disfrutar por las plataformas de Zoom y el canal de YouTube y del IBT. Pero también queremos enfatizar en este momento de manera presencial para convivir entre la comunidad del instituto y obviamente de los departamentos también. De manera presencial aquí en el auditorio Francisco Bolívar Zapata. Claro que va a haber transmisión aquí también cuando los ponentes no sean locales, bueno, que no son locales, perdón. Me permito comentar que la organización de estos simposios es un mérito de cada uno de los departamentos con apoyo de la Secretaría de Vinculación, gracias a la Secretaría de Vinculación para apoyar en esto, y obviamente también a la dirección de Laura Palomares. La Secretaría Académica realmente no ha participado mucho de la organización de estos eventos y afortunadamente me tocó inaugurar esta sesión y a su vez los simposios del 40 años del IBT por los departamentos, porque la doctora Laura Palomares en este momento está participando de la reunión del CETIC y por eso se disculpa que no pude estar el día de hoy. El día de hoy, como le decía, tenemos un programa extraordinario. Primero tenemos una sorpresa de parte del departamento donde nos van a presentar cada uno de ellos y ellas cápsulas informativas de los grupos del Departamento de Microbiología Molecular de 10 a 11 a.m. Y posteriormente tenemos varias pláticas, la primera de ellas por la doctora Linda Amaral, donde nos hablará algo de lo que llaman Plastisphere, o sea, acerca de los plásticos y los microorganismos en algunos nichos ecológicos específicos. Posteriormente tenemos algo que es como un diálogo, debate, perspectiva, una retrospectiva de la era de la microbiología por el doctor Stanley y el doctor Moseilo. Posteriormente habrá una sesión también con 2 participantes, con el doctor Monsen Gaby y Rafael Rosano, que hablarán de la resistencia antimicrobiana, estudios que tienen que ver con estos aspectos a nivel global, pero también a nivel de México, muy interesante, muy importante, yo creo que para varios de nosotros este tema, en general para la comunidad. Y posteriormente, al final, el doctor Robert Edwards nos hablará del tema de Machine Learning and Microbial Genomes. Pues realmente esta inauguración es breve para que pasemos como dice el doctor Enrique Merino en sus correos, a disfrutar de esta fiesta académica del Departamento de Microbiología Molecular. Bienvenidos a todos, ya sea presencialmente, por Zoom o por YouTube. Adelante.
fármacos son inocuos a los humanos y son biodegradables. Por lo tanto, son como la insecticida ideal. Algunas de estas toxinas, cuando se producen en la fase de esporulación, se acumulan en cristales paraesporales, como podemos ver en esta microfotografía. Y cuando el insecto ingiere este cristal, se intoxica y se muere. Algunas otras toxinas, como las toxinas BIP, que vemos aquí, se producen durante la fase vegetativa de la bacteria. Las toxinas que se comercializan y que se han utilizado durante los últimos 50 años son las toxinas CRI, las toxinas CIT y las toxinas BIP. Y nosotros estudiamos a todas estas toxinas. Los insectos, como ustedes saben, son responsables de las pérdidas en las cosechas agrícolas, y entonces lo que han hecho los agricultores es utilizar insecticidas químicos. Sin embargo, los insecticidas químicos son altamente tóxicos para los humanos. Todos ellos producen múltiples enfermedades. No existe ni un solo insecticida que no sea tóxico para los humanos. Todos tienen efectos indeseables y además son recalcitrantes, es decir, estamos contaminando el planeta. Por lo tanto, la pregunta es, ¿cuál es la alternativa? El control biológico, utilizando las proteínas insecticidas de Bacillus thuringiensis, es una alternativa viable que nos permitiría tener el control de insectos en un ambiente ecológicamente compatible.
Nuestro grupo de investigación participan 2 investigadores titulares, la doctora Isabel Gómez y el doctor Sabino Pacheco, 2 técnicos académicos, la doctora Blanca García y el biólogo Jorge Sánchez. Y tenemos 2 técnicos contratados por contrato, que es la licenciada Samira López y la licenciada Lisbeth Cabrera. También contamos con apoyo institucional del Instituto, como apoyo secretarial por Graciela Domínguez y Dagoberto Romero, quien es el laboratorista del laboratorio. Asimismo, tenemos asociados 5 investigadores postdoctorales, quienes son pagados por CONACYT o PAPIT, y varios alumnos de posgrado y de licenciatura.
Las toxinas insecticidas de Bacillus thuringiensis actualmente se utilizan como spray insecticida. Esto ya tiene muchos años de ser utilizado así y contamos a nivel comercial de diferentes formulaciones que se pueden utilizar tanto para control de plagas agrícolas como para control de mosquitos. Sin embargo, también se utilizan con plantas transgénicas. Se han transformado estas plantas, diferentes cultivos como maíz, algodón, que se utilizan donde la toxina se produce dentro de la planta y entonces ya no hay que aplicar el insecticida de manera externa. La planta es tolerante a los insectos.
El principal objetivo es entender el modo de acción de las toxinas insecticidas producidas por Bacillus thuringiensis. En particular, los mecanismos moleculares o las bases moleculares de la especificidad de estas toxinas y los mecanismos para su inserción en la membrana y la formación de poro.
Para poder definir las bases moleculares de la especificidad, trabajamos con diferentes órdenes de insectos lepidópteros, como el que se muestra en la fotografía, pero también con insectos coleópteros y con dípteros. Y el propósito del trabajo es definir los sitios de interacción de las toxinas y de los receptores. Por esta razón también hemos llegado a definir algunas de las moléculas receptoras que estaban presentes en los insectos susceptibles. Hemos hecho varias aportaciones en el grupo para el estudio de estas toxinas, entre las que podemos destacar, como ya mencioné, el poder definir los receptores, los sitios de interacción de las toxinas y los receptores, y esto nos ha llevado a entender que el mecanismo de acción es un mecanismo de acción complejo, en el que no solo las toxinas activadas son capaces de llevar a cabo el efecto tóxico, sino también las protoxinas. Estos también son capaces de interaccionar con diferentes receptores presentes en los epitelios de las larvas susceptibles, y las 2 formas, la protoxina y la toxina, conllevan a la formación de oligómeros que se insertan a la membrana y forman poros para alisar las células del intestino de las larvas susceptibles. También hemos trabajado para poder definir cómo es que la toxina se inserta en la membrana. Ha habido múltiples trabajos de otros grupos de investigación donde han propuesto diferentes modelos para la inserción de las toxinas en las membranas. Sin embargo, recientemente nosotros hemos propuesto un nuevo modelo que define puntualmente cómo es que la toxina sufre cambios conformacionales, sobre todo en el dominio 1, que le permiten insertarse en la membrana.
Asimismo, nos interesa estudiar los mecanismos de sinergismo que se presentan entre algunas de estas toxinas para potenciar su actividad, así como los mecanismos de resistencia que algunos insectos han desarrollado para contender con estas proteínas. Entre los estudios de mecanismos de sinergismo, hemos trabajado principalmente en el sinergismo entre la toxina CIT y la toxina CRI. Sin embargo, recientemente también hemos estudiado mecanismos de sinergismo entre otras proteínas, como el mecanismo de sinergismo entre toxinas BIP y toxinas CRI, lo cual permite entonces ampliar el rango de utilización de estas toxinas y generar estrategias donde todas las toxinas se potencian y se tiene mejor control biológico de los insectos. En cuanto a los mecanismos de resistencia, podemos destacar que el principal mecanismo de resistencia está ligado a la alteración de los receptores que funcionan dentro del mecanismo de acción de estas toxinas, y hemos hecho aportaciones importantes respecto a definir estos mecanismos de resistencia y cómo contender con la resistencia para sobrepasarla y poder generar estrategias de control biológico a pesar de que algunos insectos hayan desarrollado resistencia.
Ese trabajo de investigación lo hemos desarrollado de manera conjunta con diferentes colaboradores internacionales. En particular queremos mencionar al doctor Sayed de la Universidad de California en Estados Unidos, con quien hemos tenido una muy fructífera relación y hemos obtenido financiamiento como diferentes donativos, entre los que quisiera yo destacar los donativos de los Institutos Nacionales de Salud de Estados Unidos, que por 15 años consecutivos fuimos financiados para el estudio de las toxinas en mosquitos. También el doctor Bruce Tabashnik, quien es un experto en los mecanismos de resistencia, hemos colaborado para poder desarrollar toxinas que puedan saltarse la resistencia en los insectos. Y también hemos obtenido financiamiento del Departamento de Agricultura de Estados Unidos. Sin embargo, de las colaboraciones más importantes es el que tenemos actualmente, y ya por 15 años consecutivos, con la compañía Corteva. Antes conocida como Pioneer Hybrid, Corteva es una empresa transnacional que produce plantas transgénicas como algodón transgénico o maíz transgénico, y con los que llevamos colaborando ya casi por 15 años. También hemos obtenido financiamiento por parte de ellos y hemos podido patentar diferentes desarrollos de nuestro laboratorio. Finalmente, desde hace unos 8 años, hemos establecido colaboraciones con diferentes grupos en China y diferentes grupos en Brasil. Han sido colaboraciones muy fructíferas, las que continuamos en la actualidad.
Los trabajos que tenemos actualmente en colaboración con China o con Brasil se enfocan en gran medida con este insecto que se llama Spodoptera fusiperda, también conocido como gusano cogollero del maíz. Esta es la plaga principal en nuestro país, en los cultivos de maíz, que como ustedes saben es el principal cultivo de México. Esta plaga antes se encontraba únicamente en América y a partir del año 2016 migró hacia África. Y en el año 2017 ya había colonizado diferentes lugares de África y se volvió un problema realmente muy severo en esta región. Posteriormente ya pasó a India, a China, e inclusive en el 2020 se encuentra en Australia. Por lo cual en todo el mundo están estudiando esta plaga porque es una plaga muy voraz que está causando estragos y grandes pérdidas económicas.
También tenemos un interés particular en el estudio del modo de acción de las toxinas que matan mosquitos. Esto es porque los mosquitos son vectores transmisores de diferentes enfermedades. En el caso del mosquito Aedes aegypti, este es el vector que transmite el dengue, el virus del dengue, otros como Zika, chikungunya, y hemos estudiado el modo de acción de las toxinas insecticidas en este mosquito. Esto nos ha llevado a poder desarrollar algunos productos como el que mostramos aquí, que es una formulación muy eficaz que permite controlar las larvas de los mosquitos en cuerpos de agua potables y que tienen alto recambio de agua. Esperamos poder comercializar esta formulación en un futuro cercano.
Finalmente, para mencionar los proyectos nuevos que hemos estado trabajando recientemente, me gustaría mencionar que hemos iniciado con estudios de edición de genomas de insectos por la técnica de CRISPR-Cas, para poder entender mejor el mecanismo de acción de las diferentes toxinas que estamos estudiando. También hemos iniciado análisis de microscopía confocal utilizando diferentes toxinas marcadas con colorantes fluorescentes y técnicas de alta resolución en colaboración con diferentes investigadores del Instituto. Todos estos proyectos están destinados a generar mejores y nuevas estrategias para el control de insectos plaga, que sigue siendo nuestro principal objetivo.
Les agradecemos su atención Esperábamos poder recibirlos si quisieran visitar el Instituto de Biotecnología y conocer los diferentes proyectos de investigación que desarrollamos actualmente. Muchas gracias.
¿Sabías que la resistencia a antibióticos representa una de las principales amenazas para el futuro de la humanidad? En la actualidad, las infecciones causadas por bacterias resistentes a antibióticos son muy difíciles de curar. Pero, ¿qué son los antibióticos? Son fármacos, medicamentos que matan o inhiben el crecimiento de las bacterias. Los usamos para curarnos de las infecciones producidas por diferentes bacterias patógenas. Los antibióticos afectan procesos básicos para la sobrevivencia de las bacterias, como la síntesis de su DNA o proteínas o de su envoltura celular. Sin embargo, las bacterias se pueden volver resistentes a los antibióticos a través de diferentes mecanismos, como la producción de enzimas que degradan o modifican a los antibióticos, o por medio de mutaciones que afectan la unión de los antibióticos a sus moléculas blanco. Los antibióticos no tienen efecto sobre estas bacterias resistentes. Además, las bacterias son capaces de transferirse entre ellas material genético que puede ser determinante para la resistencia a antibióticos. Con esto se favorece la generación de bacterias resistentes a múltiples o incluso a todos los antibióticos disponibles. Los antibióticos no solo se usan en humanos, también en animales y en plantas. Incluso, el consumo de antibióticos en animales es mayor que en humanos. A través de los alimentos o del contacto directo, se pueden dispersar fácilmente las bacterias resistentes a antibióticos entre humanos, animales, plantas y el medio ambiente en general. Se espera que la pandemia por COVID-19 incremente la resistencia a antibióticos por el uso excesivo de antimicrobianos. Más del 90% de los pacientes con COVID-19 han recibido antibióticos. Por otro lado, se sabe que los desinfectantes o sanitizantes pueden generar de manera cruzada resistencia a antibióticos. La resistencia a antibióticos es una pandemia a la que no se le ha dado mucha difusión, pero es mortal. Solo en el año 2019 se estimó que hubo casi 5 millones de muertes en el mundo asociadas a la resistencia a antibióticos. Asimismo, se ha estimado que de no controlarse este problema, en el año 2050 podría haber 10 millones de muertes, llegando a ser la resistencia a antibióticos la primera causa de muertes en el mundo. Esto nos deja ver que la resistencia a antibióticos puede ser un problema incluso más grave que la pandemia por COVID-19. El primer antibiótico comercial, la penicilina, se descubrió hace casi 100 años. Este antibiótico lo produce un hongo. Desde entonces se han descubierto muchos otros antibióticos, la gran mayoría a partir de bacterias y hongos del suelo. Actualmente, la búsqueda de nuevos antibióticos se está enfocando a partir de microorganismos que están en el mar, en cuevas, en el desierto y en los animales, entre otras fuentes poco exploradas. En nuestro grupo de investigación tenemos la idea de que la diversidad microbiana de México puede tener un gran potencial para el descubrimiento de nuevos antimicrobianos. Por lo tanto, estamos trabajando en la identificación de estos compuestos a partir de bacterias aisladas de diferentes fuentes. Por ejemplo, del ajolote, de diferentes insectos y del mar del Golfo de México. Esto en colaboración con varios colegas de la UNAM, y de la Universidad del Estado de Morelos. Hemos aislado varias bacterias que inhiben el crecimiento de diferentes patógenos de alta relevancia clínica, incluyendo de cepas resistentes a antibióticos, lo cual hace posible el descubrimiento de nuevos antibióticos u otros compuestos antibacterianos a partir de la diversidad microbiana de México. También estamos trabajando en la identificación y caracterización de los compuestos específicos con actividad antibacteriana, lo cual nos permitirá determinar su posible uso en humanos, animales, plantas o el medio ambiente. Ahora que sabemos que la resistencia a los antibióticos representa una seria amenaza para la salud mundial, los invitamos a sumarse al combate de esta problemática. Debemos tener en cuenta el eslogan que ha usado la Organización Mundial de la Salud para advertir sobre la gravedad de la resistencia a antibióticos. Si no hay acciones hoy, no habrá cura mañana.
Hola a todos, yo soy el doctor Edmundo Calva. Aquí estoy ejerciendo otra de las formas de la cultura, que es la música, haciendo la ciencia una forma cultural muy importante. Estoy en el Departamento de Microbiología Molecular, en el Instituto de Biotecnología de la UNAM, y lo que yo me he dedicado es a estudiar la patogénesis molecular de Salmonella entérica. Tengo tiempo solo de hablarles del proyecto principal que estamos llevando a cabo actualmente, que es sobre la genética molecular de las porinas en Salmonella entérica cerevisiae typhi. Salmonella entérica cerevisiae typhi es una bacteria gram negativa que es agente causal de la fiebre tifoidea en los humanos. Las porinas de Salmonella typhi, hemos determinado que hay dos porinas mayoritarias que llamamos OMC y OMPF, y estos están reguladas su expresión de sus genes por este regulador que se llama OMPR, que está enlazado en su sistema de dos componentes AMC. Hemos encontrado que hay una regulación diferente en Salmonella typhi que E. coli, en lo que se refiere a ambas proteínas OMC y OMPF, y hemos propuesto otro modelo de regulación. También en el laboratorio hemos descubierto dos nuevas porinas que llamamos OMPC1 y OMPC2. Estas son quiescentes, por eso tienen esta Q, porque se expresan muy poquito en condiciones estándar de laboratorio y las mayoritarias se expresan de manera muy abundante. Entonces, aquí, aquí esto es un enigma, porque una bacteria quisiera tener tantas porinas y unas sean mayoritarias y otras quiescentes. Estas son los genes de los genes para las proteínas OMPC1 y OMPC2 son regulados por el regulador LEO, que a su vez regulan varios otros genes, entre ellos los CRISPR-Cas. OMPR, la propia OMPR, este regulador de las porinas, es regulada también, lo hemos encontrado aquí en el departamento, por LTRR y por algunas de las proteínas Cas de este sistema de CRISPR-Cas. Estos reguladores, de manera independiente, actúan sobre el promotor 1, pero no sobre el promotor 2 de OMPR para producirla y que regule a estos genes de porinas. El LEO antagoniza la actividad de HNS y STPA para aprender a OMPC1 y OMPC2 sobre los genes correspondientes, o puede antagonizar a esta proteína nucleóide HNS que apaga a todos esos genes. HNS y STPA apagan a los genes, y LEO es un activador porque antagoniza a HNS y STPA. hemos encontrado que puede haber fenotipos quiescentes, es decir, cuando genes que se expresan muy poquito se remueven, se quitan del cromosoma, y llega a haber fenotipos. Entonces, probablemente esas expresiones a muy bajo nivel tienen que ver con la fisiología de la bacteria. Probablemente todos estos serían fenómenos epigenéticos. Y hemos visto, por ejemplo, que OMC tiene que ver con esencias sales biliares, pero no OMPF. Y no sabemos sobre OMPC1 y OMPC2, así es que es posible que cada una de estas polinas tenga una función diferente. Todas ellas son antígenos protectores de la regulación en un modelo de Fatón, lo que nos puede indicar que todas están funcionando durante una infección. Más ahí también sabemos que la interacción entre las proteínas de membrana externa y el lipopolisacárido es crucial. Y hemos visto también que la atenuación de cepas mutadas en la OMPC1, OMPC2 y LEUO es muy relevante. Y eso también nos puede indicar que estos genes están involucrados en sus productos en la infección. También hemos visto unos fenotipos interesantes donde falta la proteína OMC y la OMPC2, o bien la OMPF y la OMPC2, y donde hay un incremento en la biopelícula. Eso podría ser un cambio en la estructura de la membrana externa. El gen LEO, a su vez, es regulado negativamente por HNS y Lrp, tiene 7 promotores, y sabemos también de manera reciente que es encendido por los genes LIA y el RHA que antagonizan el principio HNS y ALRP. En todos estos procesos, pues también participar en la RNA polimerasa, podría estar también participando en factor de terminación RO, y entonces todos estos genes, todos estos reguladores, LTR, CAS, HNS, STPA, LEUOS, LIA, LRHA, RNA polimerasa y RO, probablemente están interaccionando de maneras muy complejas y muy elaboradas para en un momento dado prender a los genes y en otro momento dado apagar a los genes de acuerdo a las necesidades de la célula. Y bueno, esta es la parte emocionante, fascinante y compleja de regulación genética en la que estamos trabajando. Muchas gracias.
Hola, soy la doctora Guadalupe Espín, jefe del grupo de genética molecular de la síntesis de polímeros y la diferenciación celular en la bacteria Azotobacter vinelandii. En nuestro grupo somos 3 investigadores, 2 técnicos académicos, 14 estudiantes y tenemos también personal administrativo que nos apoya en nuestras labores diarias. Esta es una fotografía de uno de nuestros seminarios semanales. Nuestro modelo es la bacteria del suelo Azotobacter vinelandii. Es una bacteria gram negativa que pertenece a la familia Pseudomonadaceae. Esta bacteria, como se ve en este esquema, sufre un proceso de diferenciación y forma quistes que son resistentes a la desecación y también produce polímeros de importancia industrial como el hexapolisacárido alginato, que es un agente gelificante y espesante, el poliéster polidroxialcanoato, que es un plástico biodegradable en poliéster, que es de la familia de los polidroxialcanoatos, y también produce lípidos fenólicos, alquilresorcinoles, que tienen actividad antifúngica. Como les muestro en este esquema, hemos identificado los genes de la biosíntesis de PHB, de alquilresorcinoles y de alginatos, y también hemos identificado varios genes que codifican para proteínas reguladoras de la expresión de estos genes, algunos de los cuales pertenecen a sistemas de regulación global en bacterias, como es el sistema GAC GAK, el sistema PTS-NTR y los factores sigma E y RPOS, entre otros. En el caso de la síntesis de PHB, actualmente estamos estudiando el mecanismo por el cual esta proteína E2A-NTR, que es un regulador negativo, promueve la degradación del factor sigma RPOS por el complejo chaperona proteasa CLAP. También estamos estudiando el efecto que tiene la ausencia de la proteína GAK sobre los niveles de RPOS que están reducidos, y estamos estudiando el mecanismo por el cual esto sucede. Y otro de los proyectos en el que estamos estudiando es el efecto del guanosin tetrafosfato y de la proteína de KSA, que tenemos evidencia de que cuando se disminuyen o están ausentes, se disminuye la síntesis del PHB. También relacionado con la síntesis de PHB y su producción en esta bacteria, el doctor Daniel Segura estudia la genética molecular de la degradación de este polímero, que involucra a varias PHB de polimerasas y trabaja también en el diseño y modificación de cepas para la producción de nuevos polihidroxialcanoatos. Y también en la modificación y estrategias para que permitan facilitar la extracción de los PHAs de manera simple y limpia. Respecto a los estudios sobre la regulación de la síntesis de alquilresorcinoles, estos son algunos de los sistemas que regulan la síntesis del PHB, también regulan la síntesis de estos lípidos antifúngicos, y también el conocimiento que hemos generado de su síntesis de regulación lo estamos aplicando para modificar su producción, para producir cepas con potencial para producir estos lípidos fenólicos. En el caso de los proyectos relacionados con la síntesis de alginato, de la que es principalmente responsable la doctora Cintia Núñez, estamos estudiando el papel del DCGMP, conocido también como segundo mensajero, que es clave durante la diferenciación de Azotobacter vinelandi, ya que hemos encontrado que los niveles de este compuesto varían durante el proceso de diferenciación, y también el CDGMP hemos encontrado que controla tanto la síntesis, la concentración del alginato como su peso molecular. Finalmente, quiero invitar a los estudiantes que estén interesados en incorporarse a nuestro grupo en alguno de nuestros proyectos. Estaríamos muy contentos de recibirlos y de que participen en estos proyectos y de que se formen en la investigación de microbiología.
Buenos días, mi nombre es Ismael Hernández Lucas, soy investigador del Departamento de Microbiología Molecular del Instituto de Biotecnología de la UNAM. Nuestras investigaciones se centran en el patógeno de humanos Salmonella typhi. Salmonella typhi es el agente causal de la fiebre tifoidea. La fiebre tifoidea es un problema de salud mundial, ya que infecta a 20 millones de personas anualmente, de las cuales 200,000 pierden la vida. Salmonella typhi provoca fiebre tifoidea mediante la ingesta de agua y alimentos contaminados. Esta bacteria es capaz de sobrevivir la presencia de péptidos catiónicos antimicrobianos en la boca, el pH ácido del estómago, la presencia de sales biliares y péptidos catiónicos antimicrobianos en el intestino delgado. Salmonella typhi se disemina en todo el organismo mediante el torrente sanguíneo, provocando la muerte del hospedante. Es importante mencionar que del 3 al 5% de las personas que sufrieron fiebre tifoidea, Salmonella typhi es capaz de residir en la vesícula biliar por más de 30 años. Esta es una estrategia biológica que este patógeno utiliza, ya que el residir en la vesícula biliar le permite seguirse diseminando al ambiente mediante las heces y de esta manera seguir produciendo infecciones sistémicas. Esta es una razón por la cual nuestro grupo de investigación se ha centrado en conocer los elementos genéticos que utiliza Salmonella typhi para sobrevivir en la vesícula biliar. El conocer estos genes nos ayudará a erradicar a Salmonella typhi de la vesícula biliar y reducir infecciones sistémicas. Una segunda línea de trabajo que cultivamos en nuestro laboratorio es el papel del sistema CRISPR-Cas en Salmonella typhi. Existen reportes los cuales demuestran que CRISPR-Cas es un sistema de defensa contra fagos y plásmidos en muchas bacterias. Sin embargo, estudios de nuestro laboratorio han demostrado que CRISPR-Cas es un conjunto de proteínas que regulan muchos procesos biológicos de Salmonella typhi, incluyendo la virulencia. Otra tercera línea de trabajo que cultivamos en el laboratorio es el estudio de los reguladores transcripcionales del tipo Lrse. Estos reguladores transcripcionales se encuentran distribuidos en muchas bacterias y están involucrados en virulencia, patogénesis, quorum sensi, motilidad, formación de biofilm, entre otros procesos. Salmonella typhi posee 45 reguladores tipo Lrse y estudios de nuestros laboratorios han demostrado que algunos de estos se encuentran involucrados en la virulencia de Salmonella typhi, específicamente en la invasión y replicación en macrófagos de humanos. En resumen, nuestros estudios se centran en el estudio de Salmonella typhi y de sus elementos genéticos involucrados para producir fiebre tifoidea. Estos estudios están encaminados a poder diseñar estrategias terapéuticas basadas en estudios genéticos para poder controlar la fiebre tifoidea. Muchísimas gracias.
Buenos días, mi nombre es Enrique Merino.
Y el mío es Rosa María Gutiérrez, y juntos dirigimos el Grupo de Genómica Computacional del Instituto de Biotecnología de la UNAM.
Como resultado del desarrollo de nuevas metodologías de secuenciación automatizada del DNA, actualmente contamos con bases de datos públicas que contienen decenas de miles de secuencias genómicas de bacterias, arqueobacterias, así como de organismos eucariontes superiores.
Aunado a lo anterior, la capacidad de determinar las secuencias genómicas de muestras tomadas directamente de distintos ecosistemas, como lo son las minas ácidas, el fondo del mar o el intestino de diversos organismos, incluyendo el humano, ha abierto la puerta a una nueva era de la biología conocida como era posgenómica.
Paralelamente, también se han obtenido avances tecnológicos significativos que han favorecido al estudio de proteomas, transcriptomas, interactomas, metabolomas y un gran número de ciencias conocidas actualmente como ciencias ómicas.
Con este nuevo universo de información, el objetivo central que hemos planteado en nuestro grupo es el de entender el significado biológico de la información contenida en genomas y metagenomas y de cómo esta información se genera, evoluciona y expresa.
Para ello, empleamos y desarrollamos métodos de análisis in silico para elaborar modelos biológicos que nos permitan generar hipótesis verificables que guíen nuestra experimentación a un nivel genómico.
Las líneas directrices de nuestro grupo de investigación son tres: la identificación de elementos de regulación de la expresión genética en bacterias mediante genómica comparativa, el análisis de redes en sistemas biológicos y poblaciones, y la inferencia del potencial metabólico en metagenomas.
Dentro de la primera de nuestras líneas de investigación relacionada con la identificación de elementos de regulación en genomas bacterianos, los primeros elementos de estudio son los factores transcripcionales. Mediante genómica comparativa, identificamos sus potenciales sitios de unión y posteriormente construimos modelos que representen su dinámica de regulación. Con una aproximación similar, también estudiamos a los diferentes factores sigmas alternativos y sus correspondientes promotores transcripcionales en genomas bacterianos. La curvatura estática del DNA es también uno de los componentes de regulación estudiado en nuestro grupo. Para ello, realizamos cálculos de los desplazamientos angulares y lineales de la cadena del DNA y observamos las tendencias de ciertos promotores por contener regiones de DNA puro. En relación a la regulación de la expresión génica por término prematuro de la transcripción, estudiamos diferentes mecanismos asociados a los atenuadores transcripcionales, siendo los riboswitches uno de los elementos de regulación en los que hemos enfocado nuestra atención por ser elementos de RNA que tienen la capacidad de reconocer a su molécula blanco con alta especificidad y gran afinidad sin la necesidad de intermediarios proteicos. Referente a la regulación de la expresión mediada a nivel traduccional, en nuestro grupo estudiamos las variaciones que presentan los sitios de unión de ribosoma en diferentes clases filogenéticas y conducimos estudios para caracterizar la influencia de estructuras secundarias del RNA en el proceso de la traducción. El desarrollo de metodologías computacionales para la identificación de RNAs pequeños bacterianos es otro de los temas que realizamos en nuestro grupo. Los RNAs pequeños en bacterias llevan a cabo su actividad de regulación mediante su unión a la secuencia Shan-Delgado de los RNAs mensajeros, inhibiendo con ello el proceso del inicio de la traducción.
Con las colecciones de factores transcripcionales predichos y caracterizados experimentalmente en bacterias, nuestro grupo aplica y desarrolla métodos estadísticos para integrar redes de regulación e inferir módulos funcionalmente relacionados. En el caso de Bacillus subtilis, nuestro grupo realiza también modelos matemáticos que describen el comportamiento estocástico de los procesos de diferenciación, en donde el siguiente paso es realizar experimentos que prueben nuestras predicciones. Los estudios sobre modularidad realizados en Bacillus subtilis nos hicieron preguntarnos si los componentes de los módulos de esporulación estaban conservados en todos los firmicutes. Para ello, desarrollamos métodos para la predicción de genes ortólogos lejanos que han dado y seguirán dando luz sobre la distribución, conservación y evolución de los componentes que regulan la diferenciación de este y otros destinos celulares. Hemos usado también nuestra experiencia en el desarrollo y aplicación de modelos estadísticos para definir el potencial metabólico de metagenomas shotgun. Los resultados del primer modelo se aplicaron a muestras de agua y sedimento marino del Golfo de México. El método desarrollado es de gran alcance y se aplicará a metagenomas colectados en otros ambientes. Nuestra experiencia en el desarrollo de bases de datos como la implementada para la descripción de bacterias del Golfo de México y el potencial metabólico nos permitieron responder a la emergencia sanitaria causada por la diseminación del virus SARS-CoV-2 en México, abriendo una colaboración con la Dirección General de Epidemiología para la estandarización de la base de datos CISBER. Es importante mencionar que los datos epidemiológicos colectados en las bases de datos brindan una oportunidad única para describir la evolución de la pandemia en México. En este sentido, estamos aplicando modelos estadísticos y de redes causales para entender la relación entre las distintas variables epidemiológicas y su influencia sobre la gravedad y mortalidad en la población mexicana.
Actualmente, nuestro grupo está integrado por estudiantes de doctorado, maestría y de licenciatura, así como personal académico, administrativo y de apoyo en el laboratorio. A todos ellos agradecemos su importante trabajo.
Hola, ¿qué tal a todos? En esta ocasión les voy a platicar sobre las líneas de investigación que se llevan a cabo en mi laboratorio, en el Departamento de Microbiología Molecular, en el Instituto de Biotecnología de la UNAM. Básicamente nos dedicamos al estudio del microbioma en distintos modelos animales, para lo cual utilizamos distintas aproximaciones de secuenciación masiva, como es la metagenómica, para saber cuáles microorganismos están presentes en ese nicho ecológico, la metatranscriptómica para saber cuáles microorganismos y cuáles genes están activos y potencialmente contribuyendo a las funciones de ese nicho, y particularmente la virómica para saber cuáles virus están presentes. Y de estos, claramente los que nos interesan son los bacteriófagos, para saber cómo esos bacteriófagos modulan la presencia, ausencia de ciertos microorganismos y las interacciones dentro de todo el ecosistema, porque al final son moduladores del ecosistema. Entonces, aplicando estas 3 estrategias podemos entender cómo ciertas variables como probióticos, la interacción del sistema inmune, la presencia de antibióticos, ciertas dietas, cierto estrés modula o cambian al microbioma, ya sea para bien o para mal. Mediante el uso de estas tecnologías podemos darnos una idea de lo que está pasando en ese nicho y las interacciones que se están dando. Por un lado, estudiamos el papel de la microbiota en el humano, específicamente en el intestino, en donde sabemos que hay muchísimos microorganismos, incluidos bacterias, virus, hongos y parásitos, en la obesidad infantil. Y para ello, pues, estudiamos a niños normopeso y los comparamos con niños con obesidad, y vemos las bacterias diferentes, en qué se diferencian, en qué se parecen y si tienen fagos distintos o no. De tal manera que lo que queremos ver es cómo los fagos contribuyen en pasar de una homeostasis entre todos los microorganismos allí presentes a una disbiosis, ¿no? La cual está obviamente asociada a la obesidad. Y esto te lleva, pues, a la producción de ciertos metabolitos perjudiciales que luego pasan la barrera intestinal, llegan a la sangre y entonces causan la activación de ciertos marcadores de inflamación, intolerancia a la glucosa, dislipidemia y obesidad. Es de esta manera como cuando la microbiota está en un estado saludable, se interrumpe ya sea por efectos de una dieta o de ciertos bacteriófagos causando la disbiosis, y esta se asocia a la obesidad. Para ello utilizamos pues, muestras de humanos, como se los platicaba, pero en el laboratorio utilizamos modelos de ratón para entender cómo estos fagos hacen esos cambios. Es decir, a los ratoncitos los inoculamos con fagos de humanos, de bacterias que están en humanos, y tratamos de entender ese tipo de interacciones. Entonces vamos encontrando qué fagos están asociados positivamente o negativamente con la enfermedad. Al final, pues, nuestra idea es tener una aplicación potencial de todo este tipo de descubrimientos para modular la microbiota en beneficio de las personas. Por otro lado, otra de las líneas de investigación de mi laboratorio tiene que ver con una especie en un modelo, pero de gran importancia económica para el país, que es la producción del camarón blanco del Pacífico, Litopenos-Banamey. En él también estudiamos, pues, la interacción de la microbiota con el ecosistema y cómo ella tiene que ver en las epidemias que recurrentemente ocurren en el sector. Es decir, normalmente hay un balance entre todos los microorganismos que viven dentro de los órganos del camarón, pero llega un patógeno y rompe con ese balance, o puede ser al revés. Las interacciones se rompen, favorecen la llegada del patógeno y entonces se causa la enfermedad. Justo ese tipo de preguntas son las que nos interesa abordar en el estudio del camarón, con la idea de aplicaciones tecnológicas de ingeniería de microbioma para mejorar la producción de esta especie, es decir, camarones más resistentes con una mayor productividad. Entonces, para ello utilizamos distintos moduladores de la microbiota, como son pre- y probióticos, que sabemos que interactúan con las bacterias ahí presentes, y entonces encerramos camaroncitos en granjas de cultivo y vemos cómo empezamos a modular bacterias que son benéficas para la producción del camarón. Y estudiamos, utilizando las aplicaciones de metagenómica, metatranscriptómica y virómica, obviamente cómo son esas interacciones cuando cambiamos la microbiota ahí presente versus cuando no la cambia, ¿no? Cuando llega una epidemia versus cuando no la llega. Entonces, esta es la otra gran línea de investigación de mi laboratorio, cercana a la producción del camarón y con aplicación directamente en el sector. Muchísimas gracias, eso es lo que les quería platicar.
El grupo de biotecnología marina a mi cargo se formó en el año 2015 y surge como una motivación de las tareas del Instituto de Biotecnología, que comprende utilizar el conocimiento en biología para desarrollar tecnología competitiva en colaboración con el gobierno y la industria. Todo esto orientado a la solución de problemas nacionales como es la contaminación de mares y ríos. Para llevar a cabo los proyectos de mitigación ante derrames de hidrocarburos a gran escala, realizamos cruceros oceanográficos en el Golfo de México, a bordo del barco Justo Sierra de la UNAM. En él realizamos la toma de muestras que alcanzan los 3.000 metros del fondo del mar. Los ecosistemas marinos que ahí se encuentran son una fuente importante de biodiversidad, ya que poseen características ambientales muy particulares, lo que los hace muy atractivos para la búsqueda de microorganismos y enzimas con aplicaciones biotecnológicas. Entre las principales líneas de investigación del Laboratorio de Biotecnología Marina se encuentran la biodegradación de hidrocarburos en el mar, la biodegradación de plásticos y la bioprospección de enzimas marinas.
A partir de muestras de agua de mar y sedimentos expuestos crónicamente a petróleo o sus derivados, se han aislado y estudiado bacterias capaces de utilizar el petróleo crudo como fuente de carbono. Esto se ha logrado mediante el uso de técnicas microbiológicas, herramientas bioinformáticas y análisis de remoción de hidrocarburos por graviometría y cromatografía de gases. A la par, hemos probado estos aislados de forma individual y en consorcios, con diferentes sustratos en agua de mar, agua dulce y arenas costeras, evaluando su capacidad de crecimiento, niveles de remoción y degradación. Los aislados bacterianos nos han permitido diseñar consorcios sintéticos utilizando métodos estadísticos que permiten identificar las combinaciones de bacterias más eficientes para la remoción de estos contaminantes, con resultados que van desde 60 hasta un 90%. Una estrategia que nos acerca a las condiciones que se observan en los ecosistemas naturales son los modelos de mesocosmos. En el grupo se han implementado en experimentos de remoción de petróleo ligero en agua de mar, manteniendo la eficiencia observada a nivel de laboratorio. En este sentido, nos parece de vital importancia no perturbar el medio ambiente con organismos patógenos. Por lo tanto, este aspecto también se toma en cuenta y es analizado. Para nuestros estudios, es importante conocer las bacterias que trabajamos y su potencial metabólico, para lo cual realizamos la asignación taxonómica mediante la secuenciación del gen ribosomal 16S y el uso de criterios filogenéticos, así como la secuenciación de los genomas completos, en los cuales, una vez ensamblados, se realiza la búsqueda de genes involucrados en procesos de degradación de hidrocarburos. La segunda línea de investigación se enfoca en la búsqueda de nuevas bacterias marinas y las enzimas capaces de degradar plásticos. La presencia de residuos plásticos en los ecosistemas genera un fuerte impacto ambiental afectando la biodiversidad marina y los ciclos biogeoquímicos. Específicamente, los contaminantes plásticos marinos se han convertido en una preocupación importante en los ecosistemas oceánicos de todo el mundo. Para la identificación de estos microorganismos, utilizamos métodos cualitativos y cuantitativos que nos permiten seleccionar las bacterias más eficientes en la degradación de plásticos. Existe una gran diversidad de microorganismos marinos. Sin embargo, se estima que solo entre el 1 al 5% de las bacterias son cultivables. Una forma de explorar esta diversidad bacteriana y su potencial metabólico sin tener que cultivarlas es a través de bibliotecas metagenómicas. La tercera línea de investigación está enfocada en la bioprospección de enzimas marinas. En nuestro laboratorio, trabajamos en la identificación de enzimas a partir de bibliotecas construidas con ADN ambiental proveniente del Golfo de México y del río Apatlaco en el estado de Morelos, lo cual nos ha permitido identificar actividades eficientes para la bioremediación de plásticos y otras aplicaciones biotecnológicas. Hemos explorado actividades enzimáticas dentro de las que se encuentran las poliuretanasas, dioxigenasas, lipasas y esterasas. Una vez identificadas, las clonamos y expresamos heterólogamente para posteriormente purificarlas y caracterizarlas bioquímicamente. Un ejemplo de ello es la enzima 2,3-extradiol-dioxigenasa. Esta enzima es empleada en el monitoreo ambiental. Finalmente, las lipasas y las esterasas son enzimas que hidrolizan el enlace éster de las moléculas de triacilgliceroles de cadenas carbonadas de tamaños variables. Además, en medios orgánicos pueden llevar a cabo reacciones de síntesis, por lo que el interés biotecnológico de estas enzimas radica en su amplio rango de aplicaciones, como en la industria alimentaria, en la formulación de detergentes, la producción de biocombustibles y en procesos de bioremediación, solo por mencionar algunas.
En el laboratorio estamos interesados en el estudio de bacterias enteropatógenas causantes de enfermedades gastrointestinales que impactan la salud humana. Nos enfocamos en el estudio de dos patotipos de Escherichia coli, E. coli enteropatógena o EPEC, agente causal de diarrea en niños menores de 6 meses, y de E. coli enterohemorrágica o EGEC, causante de colitis hemorrágica y síndrome urémico hemolítico. Junto con Citrobacter rodentium, un patógeno específico de ratones de laboratorio y modelo de estudio para EPEC y EGEC, para las que no se cuenta con modelo animal, constituyen la familia de patógenos denominada AE por su capacidad de inducir la formación de la lesión de adherencia y esfacelamiento intestinal en la superficie de los enterocitos del hospedero. A su vez, estamos también interesados en el estudio de Salmonella entérica cerobartifimurium, un patógeno intracelular facultativo, agente causal de salmonelosis o gastroenteritis aguda y que en los últimos años ha estado asociado con mayor frecuencia de infecciones sistémicas en el hombre. Así, trabajamos principalmente alrededor de 4 líneas de investigación con el objetivo de alcanzar un mayor entendimiento de los mecanismos que les permiten a estos organismos causar enfermedad. A dichas líneas me referiré brevemente a continuación y las cuales han seguido avanzando gracias a los esfuerzos de los protagonistas actuales que aparecerán en cada imagen y a quienes extiendo un profundo agradecimiento por su enorme compromiso, sobre todo en los 2 últimos años. En torno al proyecto Circuitos Reguladores de la Expresión de Factores de Virulencia en Patógenos AE, hemos estado interesados en entender cómo se integran, coordinan y funcionan reguladores transcripcionales específicos adquiridos por transferencia horizontal y reguladores globales ancestrales para regular genes de virulencia. Por ejemplo, EPEC posee un plásmido de alto peso molecular que codifica para una fimbre denominada BPP que permite a esta bacteria adherirse a los enterocitos formando microcolonias o agregados bacterianos, así como una isla de patogenicidad conocida como LI o Locus of Enterocide Defacement, la cual codifica para un sistema de secreción tipo 3 encargado de translocar diferentes proteínas efectoras desde la bacteria hacia la célula hospedera, que son responsables de inducir la formación de las lesiones AE y las cuales están codificadas dentro y fuera de la isla. Tanto el plasmio AF como el I, que fueron adquiridos por E. coli en eventos evolutivos independientes, codifican también para reguladores transcripcionales distintivos que regulan al operón BFP o al I respectivamente, pero también establecen circuitos de regulación recíproca y regulan otros genes fuera del plásmido y de la isla. Por otra parte, estudiamos la regulación y función del repertorio fimbrial chaperona-Usher. Las fimbrias CEU son estructuras filamentosas que participan en la adherencia a reservorios ambientales y células del hospedero, así como en otras funciones como la interacción con otras bacterias o la evasión del sistema inmune. EPEC y Citrobacter rodentium poseen 9 y 14 operones de la familia CEU respectivamente, que en general han sido poco estudiados. Uno de los principales retos para su estudio es que la mayoría no se expresan en condiciones de laboratorio, pero para varias de ellas existe evidencia de su participación durante el proceso de colonización del hospedero, indicando que se expresan in vivo. A través de un estudio integral utilizando como modelo a Citrobacter rodentium, tratamos de entender cómo, cuándo y dónde se expresan los diferentes operones fimbriales de la familia Chaperona-Usher y si estos representan para la bacteria redundancia o versatilidad funcional. El estudio del papel que desempeña en la fisiología de las bacterias, particularmente en aquellas que son patógenas, el segundo mensajero intracelular de GMP cíclico, que es producido por enzimas denominadas de iguanilatos y clasas e hidrolizado por otro grupo de enzimas conocidas como fosfodiesterasas, ha sido de gran interés. El genoma de E. coli codifica para varias de estas enzimas que modulan los niveles globales de GMP cíclico en la célula, así como su compartimentación para funciones específicas. En un proyecto liderado por el doctor Ricardo Oropesa, los esfuerzos están dirigidos a determinar cuál es el papel de las diversas fosfodiesterasas y diguanilatos y clasas de PEC en la formación de biopelícula y en la regulación de su virulencia. El genotipo ST213 Salmonella entérica cerobartifimurium fue identificado inicialmente en México a partir de un estudio de vigilancia epidemiológica realizado en 4 regiones del país y en algunos casos se asoció a pacientes diagnosticados con infección sistémica. A diferencia del genotipo ST19, responsable de la mayoría de los brotes epidémicos causantes de salmonelosis en el mundo, las cepas ST213 carecen del plásmido de virulencia. En su lugar, poseen plásmidos que proporcionan multiresistencia, así como un repertorio de genes que codifican para funciones diversas. En años recientes, cepas ST213 han sido también reportadas en Estados Unidos y Canadá, por lo que este genotipo emergente representa un riesgo de salud en Norteamérica. Así, entre otros aspectos, estamos interesados en definir las características fenotípicas que distinguen a las cepas de Salmonella typhi, Murium ST213 y cuál es el impacto del genoma accesorio y los polimorfismos cromosómicos en la biología y virulencia de estas cepas atípicas multiresistentes. El trabajo del grupo cuenta con el invaluable apoyo de técnicos y personal administrativo y a lo largo de los años de colaboradores dentro y fuera del país, así como de exalumnos y postdocs a los cuales también agradezco sus contribuciones.
Buenos días a todos. Es un placer y un privilegio para mí presentar a la profesora Linda Amaral Zettler. Ella es una microbióloga marina que trabaja en la Universidad de Ámsterdam en el Royal Netherlands Institute for Sea Research. Ella es investigadora líder de grupo, ha publicado más de 200 artículos que tienen 12.103 citas. Tiene desde el 2017 un índice H de 51. En el 2013, la doctora Amaral acuñó el término Plastisphere para nombrar a las comunidades microbianas presentes en desechos de plástico marinos en un artículo titulado Life in the Plastisphere, Microbial Communities on Plastic Marine Debris. Este artículo actualmente cuenta con 1.758 citas en Google Scholar. Como ella misma lo menciona en su página, está fascinada con el plástico, o más bien con la vida asociada al plástico, ya que el plástico probablemente tiene una influencia en nuestras vidas mayor de la que nos imaginamos. Considero que sus investigaciones sobre ecología marina y sobre las comunidades microbianas presentes en los desechos de plástico en el mar, que ella llama plastífera, son de gran importancia y espero que su plática el día de hoy inspire a nuestra comunidad de estudiantes y de jóvenes investigadores que inician su carrera en su elección de proyectos de investigación. We thank you, Linda. We thank you very much for accepting participate in this celebration of the 40th anniversary of our institute. Thank you very much. Can you please share your screen, Linda?
Could you please share your screen using the share screen option of your Zoom connection?
Yes, can you hear me now?
Yes, we do.
Okay, and can you see my screen?
Not yet. Could you share your screen?
Okay, sorry. I thought I was doing that. There we go. Okay.
Thank you.
Okay. Can everyone hear me and see my screen?
Yes.
Yes, beautiful. Okay, thank you. Sorry for the false start. I had to re-enter the room to give permission. So I just wanted to thank the organizers, Doctora Espin and Doctora Palomeres, for giving me this opportunity to speak with you today and to celebrate your 40th anniversary. Congratulations. I'm going to talk today about a topic that is very dear to my heart, the Plastisphere. And it sounds like there has already been a little bit of introduction to microbes on plastic in some of your own research. So hopefully this will be very interesting for you today as well. So the outline of my talk is as follows. I'm going to talk a little bit about plastic marine debris and sort of separate some of the fact versus fiction in terms of what we hear about in the news versus what we understand from a scientific perspective. And then I want to introduce you to what I call the Plastisphere. I'll talk about its diversity, function, and fate. And also talk a little bit about two topics that often get blurred, which are degradation and biodegradation, particularly from microorganisms. And finally, leave you with some conclusions and outlook. So first, I want to talk a little bit about fact versus fiction. And I'm drawing part of these concepts from a colleague of mine, Eric van Sevilla, who is a physical oceanographer at the University of Utrecht, which is not that far away from where I am here in the Netherlands now. And he describes some myths about plastic marine debris, and I want to elaborate on some of these myths. So one of them, and perhaps a very big one, is that there's this concept that out in the so-called garbage patches, there are floating islands of plastic of various magnitudes. But in reality, if you go to these gyres where the plastic is circulating, this is what you'll see, which is a lot of open ocean, a lot of water. And in fact, if you were to drag a net across the surface to the tune of 2,000 meters squared in this Pacific garbage patch, this is the size of about a petri dish. This is what you would find. So we don't find large pieces of macroplastic that's visible to the naked eye, but really much smaller pieces. And so this is an important concept, I think, because it's not this large macroplastic that's dominating the oceans, but much smaller pieces. Another myth I want to address is this concept that plastic is everywhere in the ocean. And I've already alluded to the garbage patches, and this is an example of a simulation of where plastic is most abundant in the ocean. And these, again, are these garbage patches or gyres. And so these are areas that are really concentrated in plastic that we find in the ocean. But on my very first cruise that I did at my new institute here now in the Netherlands, we took our research vessel from the Azores to Sicily. And what we found is that the amount of plastic, as you can see in these tubes here, is not very abundant in all places. And this reiterates the fact that, you know, there is not a lot of plastic everywhere. Now, to qualify that statement, the closer we look, the deeper we look, the more we start to see smaller and smaller pieces of plastic. But of course, that's going to depend on the size of your net. So fortunately, we think we've already started cleaning up the plastic soup, as it's called, because it's much larger, or rather much smaller than these large pieces. And there are various solutions that are being put forth. Some of them are very, very elaborate. This is an image of the ocean cleanup efforts to literally collect garbage like you would garbage in your, outside your home on the street, to try to do this in the ocean. There are other renditions of this for more coastal areas like Mr. Trash Wheel. And then things like the Great Bubble Barrier, which is actually in Amsterdam, that is meant to focus and concentrate a lot of the plastic that's out there. So none of these sort of approaches are really very effective at eliminating the plastic that's out there in the ocean, unfortunately. And for those of us that work on particularly microplastics, we know that even sampling microplastics in the ocean can be quite challenging. And these are some examples of tools that we use to study microplastics, including the sort of very traditional manta trawl that collects at the very surface. But then as we start to try to sample other parts of the water column, the task really becomes much more challenging. Things like multi-nets and high volume pumps, and even hydrocasts that collect the water in Niskin bottles, and ultimately sediment box cores, for example. Those are all tools that help us collect and characterize microplastics in the ocean. Another myth is this idea that many animal species are being threatened with extinction due to plastic waste. And while this is true, people tend to think of very large charismatic organisms like whales, but really where I think we need to be focusing a lot of attention is at the very base of the food chain, with organisms that are just only slightly bigger than microplastics, and now even nanoplastics that interact with them directly and do ultimately go up the food chain to larger organisms. And there is another myth that I'm not personally affected by the plastic soup. And for those of us that live on the coast, we see plastic marine debris firsthand. For others, perhaps they feel more far removed from the problem. But in fact, more and more we learn about plastic entering our food chain, plastic entering the very base of the food chain up until seafood and other kinds of parts of our everyday lives in terms of what we eat and what we drink, etc. And one of the intriguing parts of this puzzle that I really am interested in my own research is what role in particular do microbes play. We know that organisms consume plastics, for example, fish and other multicellular organisms. But what is largely unknown is how microbes interact with the plastic prior to ingestion, and whether they serve to make things worse or better, so to speak, in the overall exposure of organisms to plastic marine debris. And then finally, there's this concept that I can't really do anything to prevent or solve the plastic soup problem. But in fact, I think the solution very much begins with us. And this is an example of a solution pyramid. And at the very top of it, of course, is the prevention of waste into the environment in the first place. And while things like beach cleanups and physically collecting litter from the marine environment do help, they're really lower down the effectivity of addressing the problem of plastic marine debris in the environment. And only in the very bottom do we see recycling and disposal as solutions, if you will, to the plastic soup problem. So with that, I want to segue into the so-called Plastisphere. And it's very interesting, this term, if you look it up in Wikipedia, you'll get the following definition. It's a term used to refer to ecosystems that have evolved to live in human-made plastic environments. But our original concept of the Plastisphere was one where we likened it to the thin layer of life on Earth, or the biosphere. And that is that there's this little world that exists on the surface of plastic, the biofilm, where we have photosynthetic organisms like diatoms and cyanobacteria. We also have heterotrophs, such as the ciliate that you see here, and symbionts attached to that, and even heterotrophs that may in some part be using the carbon resulting from the plastic itself as a food source. So if we look at the plastics timeline, we know that plastic has been with us for a very long time, but it really wasn't until right after World War II that plastic really started becoming into a very high throughput production. And unfortunately, another timeline or another milestone rather in this timeline was in 1972, when the first report on plastic litter in the ocean was made. And so almost, shortly after the sort of really a large production of plastic started, as did plastic pollution in the ocean. And now we understand that something on the order of 10 million metric tons per year of plastic enters the ocean based on estimates published in 2015. So what are the sizes of plastic that are out there? Well, there's virtually everything from the nano up to the very large mega sizes. And this is just a scale based on the types of divisions that we see typically used in biological sciences. But you'll notice that there's a bit of a discrepancy between what we call a microplastic and what we might consider micro in the sort of scale, the metric scale. And part of that is because the pre-production pellets that are made are about 5 millimeters, and that cutoff was traditionally used to describe microplastics in the ocean, or rather in general. You can see here that the definition of nanoparticles and nanoplastic is sometimes less than 100 nanometers, but other people consider it less than 1,000 nanometers or 1 micron as well. So that definition is also somewhat blurred. So why do we care? Well, as a microbiologist, we care because the organisms that can hitchhike, if you will, on plastic can be harmful. And this was reported as early as 2003. The existence of harmful algal blooming algae, for example, were associated with plastic marine debris. In our own group, we discovered Vibrio disease-causing bacteria that attach to plastic and can be quite plentiful at times. And then finally, another aspect which is sometimes overlooked or not maybe fully appreciated is that plastic marine debris, and by this I mean not just small plastic, but even large, for example, ghost nets, can displace native species. And I don't know if I have that. No, I don't have it as an image. But one thing we have seen, for example, are organisms that are traditionally associated with things like sargassum macroalgae that are endemic to that alga, sometimes now can be found with these ghost nets that is not their natural habitat. So this plastosphere, as we call it, is really, I think, a multi-dimensional topic. There is, of course, the basic science attached to the topic, but it's really a very rich area for education and outreach. We've had many undergraduates, graduate, and even postdoctoral students working with my team to better understand the impacts of plastic and also the impacts of microorganisms living on plastic. But it really doesn't stop there. Policy topics are very much involved as well with the plastosphere. There is UN decisions being made about how plastic should be handled, whether or not biodegradable plastics are good or bad. And again, the bio in biodegradable refers to the microorganisms that are capable of breaking it down. So this is, again, entering the realm of politics and policy. And then finally, industry, of course, is very interested in how microbes interact with plastic as well, for a variety of different reasons, including, of course, when plastic is ultimately accidentally littered and or enters the environment where it shouldn't be. So with that, I want to talk a little bit about the diversity and function and fate of the plastosphere. And some of the primary questions that my group is focusing on are really still very rudimentary questions. Is there, for example, a core plastosphere microbiome? We talk about microbiomes in many different aspects, and we can think about the plastics, plastic litter is also having a microbiome. And is there some common element in the organisms that attach to plastic and live there? Secondly, are plastosphere communities phylogenetically diverse, but functionally equivalent? And finally, what is the ecological role and impact these microbial reefs have on marine ecosystems? So to try to address these fundamental questions, my group uses a variety of different tools, including tools like Raman and FTIR spectroscopy to characterize these pieces of plastic that we find in our nets and we study. We use things like scanning electron microscopy to visualize the microbes on plastic. And of course, we've been using next generation sequencing to profile the microbial communities and also interrogate their genomes. And finally, many members of the plastosphere can also be cultured. So we've been culturing many bacteria and also eukaryotes that live on plastic themselves. And I like to show this slide as sort of a conceptual framework for studying the plastosphere. And so the microplastics themselves vary in size, of course, but we try to focus on the very smallest of microplastics. You'll sometimes hear reference to secondary and primary microplastics, primary microplastics being nurdles or pellets that are the materials that plastic is made from, plastic materials are made from. And this is in contrast to secondary microplastics that are broken fragments of larger pieces. And so the sort of history, if you will, of where microplastics originate varies tremendously. And so over time, there are different physical and chemical forces that can serve to break down plastic. And this can sometimes give us a sense of how old the plastic is, depending on the kinds of signatures that are present in the plastic. But also, we can use certain kinds of methods to characterize, for example, the age of the biofilm that's present on the plastic. From the biological perspective, we also can look at the communities that are present using things like amplicon sequencing and more deeply metagenomics and metatranscriptomics processes that tell us more not only about who is present, but also where and what they're doing. Finally, we can go further and look at the ingestion, if you will, of certain kinds of plastic, primarily biodegradable plastic, using techniques like stable isotope probing to actually follow the carbon and who is eating the carbon itself. So one of the very first, I guess, principles that was established with respect to the differences between what lives on plastic and the surrounding seawater is that plastics really exhibit a highly variable community structure when compared to the surrounding seawater, regardless of location. And so what you see here in the upper graphs, and what you're looking at is a bar graph that represents different proportions of bacterial communities, of bacteria, different taxonomic groups of bacteria. And when you look at these patterns, you immediately see that the different samples here between themselves are much more diverse than even the signatures of bacterial communities that live in completely different ocean basins. And so when we first started in looking at the communities attached to plastic, we were really impressed with this because there was such a large variation in the communities present. And what we've further determined is that, and many others now, is that the geography really does make a difference. And this is a study that we published back in 2015 that showed that compared to polymer type, biogeography and the geography of where a piece of plastic was collected had a much bigger role in shaping the overall community of the bacteria that were living on it, of the plastosphere. And what you see here are biplots that show the relative similarity between the communities, and the closer these dots are to each other, the more similar the communities are. And you can see there's a really nice separation between the Atlantic Basin and the Pacific Basin, whereas if you start to compare different polymer types, you see a lot less of that integrity that's maintained. So it really does seem like geography is playing a big role in the ultimate communities that are found on plastic. So one of the things that is very commonly done more and more, I would say now, is looking at community succession when virgin plastic enters the marine environment. And this is a snapshot of a community that was taken from Woods Hole, which is where I spent most of my career as a scientist in Massachusetts in the United States. And what you see here are SEM images that show the development of the microbial community, the plastosphere, over time, and the corresponding development of the bacterial community. And of course, if for those of you who are familiar with amplicon sequencing, where you sequence these marker genes, these ribosomal RNA marker genes, you know that if you use general primers, the primers will pick up chloroplast genes as well. And so what we see is ribosomal RNA representing chloroplast plastidic organisms like diatoms present very early on in the colonization. And I think that this is really interesting and important, because I think there's definitely a bias, particularly in the microbiology community, towards biofilms being dominated by bacteria. And I like to show this SEM because what you're looking at here is the frustule, or the skeleton of a diatom, which is of course a photosynthetic eukaryote, and it's being sort of colonized now by bacteria. And so what we see in some of our work is that diatoms can really colonize as quickly as bacteria. And the bacteria are then interacting directly with these diatoms, and perhaps in this case, even causing lysis of the diatoms themselves. But let's suffice it to say that the biofilm producing organisms aren't restricted to the bacterial domain. So a few years ago, my lab published a study where we sort of tried to combine methods of visualizing the microbes in the plastosphere, but then also trying to better understand the makeup of those bacteria and eukaryotes that were in proximity to each other. So we know that one of the virtues of molecular biology is that it can tell you something about who is there. But in the absence of being able to visualize them at the same time, we don't have a good sense of how they're arranged in space and geospatially, so to speak. And so this method called ClassiFish, which stands for Combinatorial Labeling and Spectral Imaging Fluorescence in situ Hybridization, takes advantage of knowledge we know about the ribosomal RNA marker genes, allows us to build probes that target those ribosomal RNAs, and allow us to add fluorescent dyes that help us tag those organisms using hybridization methods. And when we do that against members of the plastosphere, we can now have a picture into a window into how these organisms are interacting. And I show you here the members of my team that have helped contribute to this research. Kathleen Schlunt was the postdoc and lead on this initial study applying this technique to the plastosphere. Jessica Mark Welch is actually a colleague at the Marine Biological Laboratory, my former institute in Woods Hole, and her group has really spearheaded a lot of these methodologies, first using oral microbiome and now segueing into other types of environments. So what we did was we compared communities across different kinds of marine environments, including the tropical Atlantic, the northern Atlantic waters, and also the Wadden Zee. And what we see is that there's quite a bit of variation in so to speak, the colonization, early colonization of biofilms. And you can sort of contrast some of the types of organisms that were present. And you'll see in the Waddenzee, which is right outside my doorstep here in the Netherlands, that the colonization was really much less than some of the other locations that we applied this technique for. So, as I mentioned before, we use sort of multi-omic methods to study the plastosphere and the organisms therein. And of course, a very powerful tool is to use genomics to characterize the genomes, look beyond who's there and look at their functional potential. We also employ metagenomics as well. And this method is applied to these individual pieces of plastic to, again, ultimately try to reconstruct the genomes of the members of the communities in situ. And so when we've started interrogating some of these metagenomes in particular, one of the things that we find, and this is an image that shows effectively the cross-section of the different genomic bins that are present within a series of different samples, libraries in this case, that have been co-assembled. So where they share similarities, you can see these bands here, and this outer ring represents the different genomes that are present. And while there's a lot of diversity that's present, one of the sort of striking aspects of looking across different samples is you start to see organisms that are biofilm formers, which isn't perhaps that surprising, but I would say that these biofilm formers are perhaps part of this core microbiome that is present on most plastic. And if we dig deeper, we can, in some cases, really reconstruct large parts of these genomes, which of course is quite exciting and helps us better understand the functional diversity and not just the phylogenetic diversity that's present in and among these communities. Okay, so now what I'd like to do is segue a little bit into talking about degradation versus biodegradation, because I think one of the things that is clearly very much in the headlines these days is how microbes can help us solve the plastic problem, the plastic pollution problem, right? And so we're probably very familiar with some of these articles I'm showing you here. And one of the things they share is this description of biodegradation, right? They've all got reference to biodegradation. But when people ask me, are microbes biodegrading plastic in the ocean? I'd like to sort of explain that the concept of biodegradation is really quite a specific one when we're talking about the impacts in the environment. And what that definition entails is really taking these very complex polymer chains and not only breaking them down into smaller fragments, but really breaking them all the way down into CO2, water, and cell biomass. Okay, this is really what biodegradation is meant to be about in the marine environment. And so when people ask, you know, is bioplastics a solution to the problem? It's not a straightforward answer. And part of that is because bioplastics is a term that is somewhat ambiguous in the literature and also effectively as a concept. And that's because bioplastics can both be bio-based, meaning that the feedstock that plastic comes from is organic in origin, is based on biological feedstock. But they can also be fossil-based, okay? So the term bio-based refers to the feedstock, whereas biodegradable refers to the end of life. So bio-based is the beginning of life, biodegradable is the end of life. And we have bioplastics that really fall into these different categories. So it's much better to speak about bio-based and biodegradable plastics when we're talking about plastics that are friendly to the environment. And it is really important to beware because there are a lot of plastics out there that have additives, but they are in fact just conventional fossil fuel-based plastics that are both fossil-based and non-biodegradable. And they have additives in them that make them disintegrate, but simply make them produce microplastics or nanoplastics that are not readily digestible by organisms, and they do not biodegrade. So the marine environment in particular poses a lot of challenges. And this recent definition from a SAPEA report describes plastic degradation, or defines plastic degradation as the extensive conversion of polymer carbon to CO2 under oxic conditions, or CO2 and methane under anoxic conditions, and new microbial biomass over a specified time frame. And I think that last phrase, over a specified time frame, is something that sort of gets lost in the literature. It's not enough to biodegrade, to have a 1% or 2% level of biodegradation. That's not biodegradation in the end. It needs to be a reasonable amount of time, not centuries, but days or months, and certainly, not decades. So some of the challenges that the marine environment poses with respect to biodegradation includes, compared to, let's say, a composting environment, limited microbial activity, high hydrostatic pressure, low temperatures, and sparse nutrients. These are all challenges present. So in my own group, we have specifically been looking at marine biodegradation, and I was fortunate to be able to obtain part of the original biodegradation community that was used to develop something called the Marine Biodegradation Standard from the ASTM International, which is D6691. And we recently published the genomes of the members of the consortium that were still intact, and help better describe some of the depolymerase activity present in some of these members. Since then, we've been working with ISO standards to better characterize the communities associated with some of these new standards that are coming online, that are geared at helping us better characterize some of the new materials that are being produced that are supposedly more environmentally friendly, and our tests that show how these materials behave in a certain period of time. And some of the older standards have a marine biodegradability requirement of at least 60% biodegradation, which means, again, evolution of via CO2 evolution in 180 days. This has been a threshold that has been suggested in some of these standards, and it's one that we've been testing in terms of looking at some alternative kinds of methods for using natural inocula versus defined inocula, and better understanding the communities that are biodegrading in the environment, particularly the marine environment. And I just want to shout out to credit my student, Fons de Vogel, who is working on better characterizing and understanding the communities that biodegrade, biodegradable and compostable plastics in the marine environment. So with that, I want to talk a little bit about some conclusions and an outlook. So some of the conclusions I'd like to draw from what I've said so far is that microbial populations colonize plastic marine debris quickly, and they can be dominated by diatoms in just 1 week after immersion. Very few taxa appear to be members of a shared plastosphere or core microbiome, but among them are biofilm formers. But the biofilm formers themselves are not necessarily always the same players. They can include different genera of bacteria, but importantly, they can also include eukaryotes. And specific eukaryotes include the diatoms, for example, that can form biofilms and attract bacteria to themselves as well. And finally, using comparative genomics with genomes recovered from metagenomic bins and binning methods will help elucidate not only who's there, but also their functional potential in these underlying communities. So because I have a captive audience, I like to finish my talks with 7 take-home messages. So I started this lecture by introducing you to 7 myths. And so I'll end it with talking about 7 take-home messages. And I was trained as a biological oceanographer. And I know we sometimes talk about the oceans, but really the ocean is one huge body of water and it's connected. And I like to say that we all share this ocean. And despite having exclusive economic zones, these EEZs, marine debris doesn't respect those boundaries, right? So we know that sometimes the litter that ends up on our beaches is, you know, we are sometimes responsible as residents, but sometimes the garbage, the litter comes from elsewhere. And, you know, this is something we need to be aware of and acknowledge that we all share this great ocean that is a source of energy and life that we all benefit from. When we think about plastic, out of sight shouldn't be out of mind. And again, I think I've illustrated quite importantly that most of the plastic that's out there, not in terms of biomass or volume, but in terms of numerics, is microplastics and nanoplastics. And so just because you can't see it doesn't mean that it's not a problem. Sort of the age-old, I think, mantra, reduce, reuse, recycle is really important, but reduce and reuse should really be emphasized over recycling. Recycling is valuable, but it does consume primary plastics and requires that. And of course, the extent to which we recycle is not enough to help sustain the environment and our lifestyles effectively. Fourthly, biodegradable compostable plastic is a solution to the plastic usage and consumption problem, but it's not the solution. Again, we have to come up with alternatives to single-use plastic and just how we approach our use of single-use plastic in general. And replacing all single-use plastic by biodegradable plastic and compostable plastic is certainly not a solution to everything. Fifthly, proper waste infrastructure and composting facilities are required for biodegradable plastics to be effective, right? We can't just have biodegradable plastic and not dispose of them appropriately so that they can then enter the circular economy and be recycled effectively and composted as they should be in order to be really full cycle. And along those lines, marine environments are not composting facilities. So we shouldn't be dumping biodegradable, truly biodegradable, and even marine biodegradable plastics in the marine environment. We should never be littering the marine environment, right? Because that's not what we should be doing in any environment. And finally, being very heavily involved with the standards community, I want to emphasize that global problems require global cooperation and collaborative efforts across international standards organizations. And sometimes this gets forgotten and left behind. While there are different standards organizations, the coordination between them is not always ideal. And I think we need to come together specifically on this topic in order to achieve the highest standards with which we measure products that are safe or unsafe for our environment. So with that, I want to thank you for your attention. And yeah, acknowledge the members of my team and collaborators that have contributed to this research, and also my funding bases that have helped facilitate the work that I shared with you today. And of course, thank all of you for giving me this opportunity to speak with you today. Thank you.
Thank you very much, Linda, for such an excellent
Are there
for
questions?
such an excellent talk. And now we are open for questions. No questions? I have one question.
Okay.
Linda, do you think, you envision the evolution of bacteria or microorganisms that can degrade plastics, non-degradable plastics? Because I am convinced that we still haven't found on, maybe there is not bacteria or bacterial consortium that really biodegrade plastic to CO2 and water, whatever. But do you think evolution will give us a consortium that degrades plastics?
Yeah. So I think we don't want to wait for this, to be honest. I think it's going to take a really long time. I mean, we know, of course, that from what we can find in the bottom of the ocean, that, bacteria are not very efficient at biodegrading fossil fuel-based plastic. Clearly, there are types of plastics that are marine biodegradable, and people are looking into those. But in terms of the plastic that's currently there, we have a big problem because it's not going away very quickly. And that's not to say that, given millennia, it's not going to go away, but that's not the point, right? I mean, we don't want to wait. We don't want to continue for that to be the norm in our environment. So yeah, I mean, these carbon-carbon bonds are very strong. And, I come from an institute here in the Netherlands, the NIOS Royal Netherlands Institute for Sea Research. And one of the big areas of focus are biomarkers. And these are lipid fossils. And the reason we can study them and use them is because carbon-carbon chains are really, really resistant. They're recalcitrant and they last a really long time. So yeah, it's been a long time, and many organisms still can't degrade those naturally occurring plastics. So yeah, I mean, I think that waiting for microbes to degrade fossil fuel-based plastics, I think, is something, an effort that we don't want to sort of look towards as a solution.
Thank you.
I have one question.
Yes. Let me read a question from Ramses Gallego. Do you think the use of GMOs, bacteria mainly, is a plausible way to help with the biodegradation of ocean plastics?
So by ocean plastics, are you referring to fossil fuel-based plastics or biodegradable plastics? I mean, I think that it's going to be difficult for regulatory agencies to approve GMO release into the marine environment. And I think that, that solution, while scientifically it's intriguing and interesting, I think the missing part of that is, the way that many of us study the bacterial function, right, is in our laboratories. We don't actually know what happens in the wild. And I think that the interactions between microbes in the wild is very complicated. And while things might work beautifully in a laboratory setting, once you get out into the wild, so to speak, things might not be so straightforward. And so I'm definitely, intellectually intrigued by the possibility of bacteria being capable of doing this. But I think the hook is, what are the timeframes in which this can really happen? And do we really, is this really the solution to the plastic pollution problem? I don't think so. I really don't think so. And I don't think that, yes, someone says, I'm not sure what the yes is behind the question that I just tried to answer was, but anyway, that's my opinion based on what I've heard and seen, and even honestly, just working with biodegradable plastics and seeing the differences between different consortia behave on biodegradable plastics. Yeah, it's complicated. It's complicated.
Thank you. Dr. Calva has a question.
Yes. Can you comment on any efforts towards lab-directed evolution? I mean, do people cultivate bacteria or other microorganisms in the presence of different types of plastics and try to select faster growers, or, are people doing that kind of work?
Yeah, for certain they are. I think that a lot of what we've learned about conventional plastics and microbes that seem to be facilitating breakdown are that they're eating these smaller molecules that are leaching out of plastic. Also it is a bit complicated too with plastics in the environment, right? Because when we study them in our lab, we give them a sole source of plastic and we see kind of how they interact with it. And when you get into the environment, things are more complicated, but they're also interesting in that, there's UV, there's mechanical wearing and tearing of plastic. So it is interesting from that perspective, I think, what microorganisms get offered, if you will, in terms of plastic, byproducts, so to speak, whether it be additives or smaller leaching molecules. And we know that a lot of stuff does leach out of plastics, and a lot of those smaller molecules are very edible by bacteria. So I think we have to, what's the end game, right? Because a lot of plastics can be recycled much more efficiently than enzymes that can be manufactured and produced. I think where things maybe we can take advantage of certain kinds of microbes and their novel enzymes is in more of the recycling kind of area, where there are certain kinds of processes that are difficult to carry out in a more cost-effective way. But even then, recycling isn't as efficient and isn't as cost-effective ultimately as we want it to be, right? I think that it has its limitations, and that's my answer to your question.
Okay, thank you.
Thank you.
We have another question here.
I know Liliana's had her. Is that okay? Is that the next question? Yeah.
Yes, thank you.
Thank you.
Hi, Linda. Thank you for your talk. I really enjoy it. So we always think about how bad the plastic are in the ocean, mainly in the transportation of pathogenic species like Vibrio. But maybe not everything is so bad. I don't know. What do you think about the role do microbes play in nitrogen or carbon fixation?
Yeah. Right. So I guess the way I look at it is it's a perturbation, right? Because as we're adding plastic to the environment, particularly the open ocean, there aren't a lot of substrates out there, right? It's kind of it's an aquatic desert, so to speak. And so I think we have to think about, okay, what's the impact that change is making on our ocean, right? The ocean is huge. It's a large volume. It affects climate. There's so many implications, right, to changing the ocean. So I think that's important to keep in mind in terms of impacts and how that translates into good or bad. I think the point is we don't know, right? And so while there could be benefits, I think the research is showing that there are changes, and change isn't always good in this case. I mean, obviously we don't have much of a choice at this stage because we're not going to effectively clean up the plastic that's there. So the best we can do is prevent it from continuing to be introduced into the ocean as, as quickly and as to the extent that it has been over the last, 60 to 80
Thank
years.
you. We have another question here in the auditorium.
Okay.
Lucia.
Thank you, Linda. Thank you for your very nice and interesting talk. And I have just one question. You said that the solution is not using biodegradable plastics, but how can we move, for example, to stop using plastic bottles? That I know that we have been moving because we have stopped using, for example, plastic bags, but not plastic bottles. I mean, chlorine comes in plastic bottles, water, and all of those liquids. So how can we move to another kind of containers that are not made out of plastic?
So I didn't mean to say that biodegradable plastics are not a solution. I completely agree that biodegradable and compostable plastics, if there is the infrastructure to compost them as they should be composted, I think that they're absolutely part of the solution to reducing the amount of plastic that ends up in the environment. I do believe this. I'm sorry if I misled you in that respect. But what I think is important to keep in mind is that there are materials that are being described as being biodegradable that are not in fact truly marine biodegradable or biodegradable and compostable in general. And these are materials that, are perceived as being safe for the environment by the general public, not really understanding what biodegradable really means. And that is a danger, right? Because now you're kind of endorsing or, encouraging even potentially people to litter in the environment, and you certainly don't want that. But surprisingly, sort of the circular economy is very critical of biodegradable alternatives to fossil fuel-based plastics, because there is this effort of recycling and having that be the part of the circular economy. I think that's slowly changing. Perhaps also because of our reliance on fossil fuel based economy. But, right now still the infrastructure to produce biodegradable and compostable plastics and also the cost of producing biodegradable plastic is so much greater than fossil fuel based varieties that they can't compete easily at this stage. I hope and expect that will change in the future. But right now, we're still in this awkward period where we're trying to embrace new polymers, new materials that will be more environmentally friendly in some way, whether it be in terms of being compostable ultimately, or in terms of, using a bio-based carbon source as opposed to a fossil fuel-based carbon source.
Thank you. We have another question here.
Hi. Thank you for the nice talk. I find it interesting the presence of photosynthetic eukaryotic organisms on the surface of these plastics. Mm-hmm. What are they doing there? Because I understood that they colonize fast the plastics. Mm-hmm. But maybe they are there just because many plastics float on the surface of the sea, and attaching there, it's useful for being exposed to the light. Do you think they are doing something or on biodegradation of plastics?
Thank you for your question. I guess I didn't mean to imply that they were necessarily doing something specific to the plastic, although ultimately, by colonizing it, they are changing the communities that attach to plastic. The kinds of diatoms that we see are ones that are typically found in benthic communities. So they are colonizing surfaces that are available to them. You know, I think that diatoms can produce materials that attract bacteria that can degrade and biodegrade chitin and other kinds of carbon-carbon molecules that are, you know, more recalcitrant, let's say. So ultimately, they could facilitate some of the fate of the materials that are contained within the plastic itself. But yeah, they're opportunists like the you know, bacteria that colonize plastic. And I think you're absolutely right. They're using a surface, and especially with plastics that float in particular, they're taking advantage of that. Yeah.
Thank you.
Yeah, thank you.
Thank you. I have views, and I have the last question. Have you, do you know, or have you make the microbiome of not floating plastic, but those that are on the soil of the sea, and how it compares?
So we have, we've started looking particularly at so the bio-based and biodegradable plastic types like PLA and PHA types of plastic do sink. I didn't really talk about this, but the predominant plastics, fossil fuel-based plastics like polypropylene, polyethylene float. And that's a majority of what you see, of course, when you look at the surface. But if you go to the depths of the ocean, you do find like PVC and polystyrene, non-expanded polystyrene. You do see that at the bottom of the ocean. We've been spending most of our efforts with floating plastic, but where we have looked at the microbiomes have been biodegradable and compostable plastics. And those communities are interesting, of course, because there I mentioned in my talk that the plastic type doesn't seem to have as big an influence on the communities as geography. But when you start to get into the biodegradable and compostable plastics, that's when you start to see some different kinds of communities, even in the same geographic location. And again, I mean, the implication there is that these plastics do provide a source of carbon that is edible, so to speak, to the more available to the bacteria themselves.
Oh, thank you, Linda.
You're very welcome.
Thank you very much for your excellent work, and we pass to the next one.
Okay, thank you very much, and congratulations on your anniversary. Okay, bye-bye.
Hello.
Hello.
Hello, Stanley.
Hi. Can you hear me, Edmundo?
Yeah, we can hear you quite well.
Let me share my screen.
Yes. Let me. Elio is. I think he got off his chair.
He should be back in just a minute.
Okay. So I'm going to. You can leave your screen on. I'm going to just say a few words about you and Elio as a very brief introduction. Then we'll go for 45-50 minutes as we talked about, so we can leave some time at the end for questions. I just want to say briefly that Elio and Stanley Malloy, well, first of all, are very good friends. And aside from being very good friends, there are two scientists that I admire, and I admire them because they're not only outstanding researchers, investigators, but they're extraordinary professors, teachers of microbiology and of science as a whole. But they also are very interested in science education to the general public. They're interested in the international issues of science. They're very sensitive
to to science as a global enterprise. And they're very complete human beings, and from my point of view, and that's why they are admirable. And so with no further ado, they're going to have a set of reflections. Go ahead, Stanley.
Ilio, you want to kick us off?
Okay. Les quiero saludar en un idioma común, pero lamentablemente no creo que todos los participantes sean hispanoparlantes, de manera que tengo que cambiar al otro idioma. So hello, everybody. What I said in Spanish.
Muy bien, Elio. Adelante, Elio.
You can hear me? Can you hear me now?
We can hear you well. Yes, we can hear you well.
I said my speaking a little bit of Spanish indicates that I think everybody should learn Spanish. Anyhow, we're back. Stanley and I were here 15 years ago on the occasion of the 15th anniversary of this wonderful institute, and we're back. So we're going to, can you see the slide? Is the slide on? Can you see the slide?
Yes, the slide is on.
Okay,
yes. Yes, we can see the slide.
Okay, let's go to the next slide.
Okay.
How do we do that?
There we go.
There we are. So this is what happened 15 years ago. Where are we? And so there was a colloquium of the American Academy of Microbiology that dealt with just that question. And we wrote an article in the journal International Microbiology that's based in Barcelona. The era of microbiology, we called it a golden phoenix. And this was the 15th anniversary of the Instituto de Biotecnología. We also have a site called This Week in Microbiology, and as well as a blog called Small Things Considered. In the next slide, what we all can agree on, I think, is that microbiology is a central discipline. It started with the discovery and integration of the science by Pasteur Koch and the students. And then as science and microbes became tools for the development of molecular biology. I'm going to make some predictions for the next millennium. So what have we learned since 2007? The future is bright. How's that? I'm now very optimistic. So the previous eras of microbiology look something like this, if you like, this kind of curve. There was a great ascendancy of the science with Pasteur Koch and the students in the end of the 19th century. It kind of slowed down when biochemistry sort of started and kind of seemed to take over. But when molecular biology started in the late 1960s or thereabouts, it was very much based on microbial work. And then eukaryotic molecular biology took over because it turned out that it was possible to study eukaryotes as well as bacteria and phages. So it went into various stages. And if you don't mind, here is a cartoon that indicates the position of microbiologists. So microbiologists are obviously small biologists. Okay.
Ilio, could I add a story here? This relates to when there was the shift from molecular biology and thinking about microbes to be able to apply those tools to eukaryotes. at that time, I was applying to graduate school and I wanted to work on bacteria. And I recall one of the interviewers saying, why don't you want to work on eukaryotes? Nobody cares about microbes anymore. That's passé. It's never going to be important again. And so it's not just microbes are small biologists, but I think that they were thought of as not being as important as other biologists.
That's right. That's right.
Okay, anyhow, so with the advent of studies, the application of studies using especially metagenomics to study what is where, what organisms are where, and using genomics to do any tremendous development in microbiology. So it's in the ascendancy right now. So why did we anticipate that this would happen? We were very smart, that's why. Okay, so the impetus was antibiotic resistance, bioterrorism, the emergence of infectious diseases, recognition that infection caused chronic disease. We're going to come back to all these points, by the way. Climate change and oil spills, the roles of microbes in natural ecosystems, and the impact of microbes everywhere. With the new tools like genomics, metagenomics, environmental probes, single cell analysis, that is, you can look at bacteria, not just a little bags of something that is very complex and very sophisticated kind of cells and metabolic support. Right?
So here was the basic idea that Elio and I thought about at that time, right? So we thought people had been focusing strictly on bacterial cultures, pure cultures of bacteria. And we had learned so much from studying those pure cultures. But it was very difficult to study complex populations before we had these tools of metagenomics and environmental probes. I think that Linda did a really nice job of emphasizing the importance of those tools. And also, there are times when a single cell can tell you more than a group of cells. And we didn't have very good approaches to study single cells. So if you looked at the period in 2006 when we wrote the paper, when you look at populations, when you have different types of microbes mixed together, with the microbial populations, and understanding the microbial diversity was hard because 99% of those microbes were uncultured. So we really didn't know much about them. In 2007, people just started getting success in some initial approaches using dilution to make single colonies. And then in 2002, we're in a very different world. From the genomes, we can make predictions about what we should use to help grow the bacteria. We understand that co-culture is sometimes important. We can use fluorescence-activated cell sorting. And we can continue to use dilution approaches like membrane diffusion and microfluidics. So we're starting to be able to culture many of those microbes that we couldn't study before. So the other question you can say is, in a population of microbes, who is there and what are they doing? In 2006, we had 16S ribosomal RNA. That could tell us something about taxonomy, but it really didn't tell us about metabolic potential of the organisms. In 2022, the ability to do real wide-scale DNA sequencing totally changed that, because genomics and metagenomics allow us to not only understand taxonomy, but to make predictions about functionality. Not to tell us what functions were there, but to make predictions. When we think about genetic exchange, which is really important if we're going to tie genomes together, in 2007, we thought it was really pretty limited. We knew it happened, but we didn't see it as being something that was sustained, because we thought whenever genetic exchange happens, almost always there's a fitness cost.
The reason
2000.
the lab, it was limited.
Right.
So
just because we could make it happen in the lab didn't mean that it was going to be the same in nature, right? So the other thing is, in 2022, actually very recently, people have been able to show that in fact genetic exchange is really rampant. And the way we think about genetic exchange and the way we think about understanding genomes is much more complicated than it was even in 2021. So metagenomic sequencing came along. In 2007, we got our initial cases. We could look at a very simple population with 10 different kinds of organisms. This was the acid mine drainage. We really couldn't do much more than that because reconstructing the data was just not possible at that time. In 2022, we can now do a lot of reconstruction of very complex microbiomes. Not everything, but we're beginning to get there. And then if you look at omics, transcriptomics, metabolomics, all of those other tools, in 2007, it was very limited. And in 2022, we can do it at single cell resolution, right? So there's been a big change over this period of time when you look at populations of microbes. Now, also with single cells, single cell analysis is really important because some processes get obscured when you look at population averages. In one cell, something might be highly expressed, in another cell, slowly expressed. You take the average, it looks like it has some medium level of expression. But if you study single cells, you can find out what that kind of variation is. In 2007, we could do very little of it. In 2022, we've been able to make single cell droplets that allow us to study those single cells and what's going on in them. If you look at a single bacterium in 2007 with DNA sequencing, it was difficult to get a good sequence. It required a lot of work with multiple replacement amplification. In 2022, it's pretty straightforward with whole genome amplification. If you think about using fluorescence activated cell sorting to get these single cells separated, in 2007, there is limited ability. In 2022, we have really strong dual reporter probes that allow us to separate them nicely. And if you look at imaging, yes. And so imaging, the importance of imaging, I think Linda really indicated, right? In 2007, you could look at populations by doing autoradiography if you could get radioactivity into them. You could do fish analysis, look at simple kinds of populations, and limited resolution cryotomography. But in 2022, we now have super resolution imaging. We've got probes that are really good. You can use them in single cells. You can see how molecules are moving in single cells. And we can study the molecular machines in single cells by high resolution cryo-EM.
Well, it sounds like we've made progress, haven't we?
Yeah, so things have come along a long ways. So let's just say, well, you know, what did Elio and I predict, and what have we learned, right? We've learned, we predicted that we would get a lot of insights into population biology, cell biology, physiology, and genetics, and we've learned so much about it, right? Our ability to study things like plastic degradation in 2007 was very limited. Now it's pretty sophisticated. When you think about understanding microbial ecology in complex environments, this question of who is there and what are they doing, we've improved there, but we have a long ways to go. We can find out who is there very well, but what they're doing has been more complicated. When you look at the manipulation of microbial communities, changing some function and seeing what happens, we're kind of there, but we have a long ways to go. We have a long ways to go because we can add things to populations, but it's very hard for us to just knock out one part of a population. one kind of cell or a particular gene, right? So we have a long ways to go now. And another thing we predicted was that a lot of these things are getting so complicated that we're going to need special centers that have certain kinds of equipment that can do things like genomics and functional genomics that many of us couldn't do in our own lab. And that we really need improved tools, better culture methods, better genome assembly from metagenomics, better probes and improved imaging. And that has really come along nicely, as you saw in the last slide. And fundamentally, we predicted, and this has really been true, that microbiology touches every other area of science. And we'll go through some examples of this. But back in the age of molecular biology, people thought of microbiology as being a little separate discipline, not something that would affect things like eukaryotic cell biology. And now we know that's the furthest from the truth. Elio, you're on.
Here's an example of what we're talking about. How do bacteria influence the weather?
Elio, just can you speak a little closer to the microphone, please?
Sorry.
Please.
Better?
Thank you.
Is this better? Is it better now?
It's better, yes.
Okay. So this is a schematic about how bacteria influence not just the weather, but the climate. The climate is a bigger concept than the weather. Anyhow, dinoflagellates make a compound called dimethylsulfopropionate. This is changed by bacteria into dimethyl sulfate, and dimethyl sulfate, as you can see on the top, is broken down by light into sulfide and sulfate. The sulfate aerosols make four clouds. So phytoplankton makes DMS, bacteria changes into DMST. This becomes sulfate aerosols, which again into the atmosphere, leads to cloud condensation and rain. So it all started with bacteria modifying DMS into DMSO. It's an example, and this is done on a very large scale. So this is an example of how bacteria in the oceans lead to a change in the weather and therefore in climate.
Elio, can I add something? To me, this is mind-blowing. If somebody in my general microbiology class when I was an undergraduate said that microbes influence whether or not it rains, I would have said, you know, that's crazy, right? But this is where microbes are impacting weather and atmospheric sciences, right?
Yeah. In other words, if you can't do weather forecast without being a microbiologist. Something like that.
So one of the other things that's been true for a long time is we could use satellites to understand phytoplankton blooms of toxic algae, a number of different things. This is a great example off the west coast of Mexico, where the redder an area is, the more chlorophyll is in the water, and you can actually see the difference between August and December. In December, you get more rain, more storms, there's more runoff into the water, and so you see more red along the coastline. That red influences the amount of oxygen in water and influences the survival of fish. So it has a really important problem. We've been able to study this by satellite to look at large areas. We've been able to do that now for a while. One of the other things that we've learned in the last couple of decades is when you think about emerging infectious diseases, there's this One Health concept where new human diseases often arise from animals, where the disease is transmitted from the animal to humans, and often they come to animals from the environment. So humans disrupt the environment, the disease is transmitted to animals, animals come in contact with humans, transmit the disease to humans. This is the fundamental principle of spillover and how most emerging infectious diseases arise, right? But like in the case on the previous slide, when we were trying to understand these algal blooms, in this case, the question is, where are these coming from, right? And we now, over the last couple of years, have been able to take technologies that allow us to study very localized places over real time to understand where things are coming from. It applies to toxic algal blooms, emerging infectious diseases, anything else you want to assay in the environment. That could be microbes that promote or harm agriculture. So the big thing here is you use GIS to locate precise spots.
What GIS means?
This is Geographical Information System. So this is a computational approach that allows you to very specifically find a specific site, right? This is Google Maps for microbes, okay? So you can find that site and go back to that site precisely every time you want. And you can go back to it using drones that can sample a particular site, take that sample from an area that may be difficult to work in, but somewhere very close, and in the field, do things like metagenomic analysis to see what's there, what the changes are, to assay for a number of metabolites.
What does min-ion mean?
This minion is a kind of sequencing. This is a technology for sequencing that's very easy to do in the field. Okay. Thanks, Elin. And then along those lines, another thing that's really been a key lesson over this time is the role of the microbiome in health and disease. I mean, we all know that there's this sense of mutualism where the host provides microbes this environment with temperature and nutrients that allow the microbes to go. And for a long time, we've known that the microbe also provides vitamins and nutrients to the host. It helps host development. So this is the interface with developmental biology, development of the intestine, development of the endocrine system, development of immunity. We know that the microbiome helps decrease the sensitivity to pathogens, in part by competition. But what we've really understood in the last few years is that the microbiome makes key substrates that are modified by the host, by us humans or animals, for key purposes. So for example, the microbes make certain substrates that are used to make key bile salts that are really important for our digestion. So the microbe substrate moves up to the liver. In the liver, it's modified, and then it comes back to the intestine. Okay? The other thing is the microbe's interference with drug function. One of the early studies on this was microbes converting acetaminophen into a toxic substance. There are certain people that acetaminophen has really bad side effects for, and it's closely tied to the microbes that are in the intestine that make certain chemical modifications to it. And more recently, it's been discovered that microbes in our microbiome, in our gut, can modify anti-cancer agents and inactivate them. So doxorubicin is a very effective anti-cancer agent. In this form, it's active. It can get modified by acylation by microbes in the gut, becomes inactive. So people who have a microbial flora that's very effective at this inactivation are less effective at being treated with that particular anti-cancer agent. And we know that microbes not only cause acute disease, but can cause chronic diseases.
We think.
We know in some cases. Let me get there, all right, Helio. So look, we know that microbes can cause cancer. There's really strong evidence for that. We know that microbes can cause heart disease. Again, very strong evidence for that. We know that microbes can cause liver disease. Hepatitis leading to liver cancer is really an old example of that. We know that microbes can cause kidney disease, right? Very often, those effects, with that exception of hepatitis that I mentioned, very often those effects are because of multi-types of microbes in a population, right? But here's the problem, and this relates to what Ilio said. There's a whole bunch of other diseases that people attribute to the microbiome, but there's not good, solid evidence for it, right? So the reason we know about causing cancer is because we can do specific things to manipulate mice to make this happen. We can do specific things with heart disease. We can do specific things with the microbiome in liver disease and in kidney disease. We can reproduce the phenotype we expect that's there. But in these other cases, we don't have an easy way of testing. And whenever you have multi-types of microbes that have to work together to do something, it's so much more complicated to understand. So we've learned a lot about these chronic diseases since 2007, but there are still so many questions. You want to take over, Ilio?
Sure. So not only do microbes cause disease, but they're also able to influence the psychology of the host. So a great example is that rats which are infected with toxoplasma lose their aversion to cat urine. In other words, they're no longer scared of cats or of little, what is that, a tiger? So that is a very big change in the psychology, in the behavior of animals. There's a virus infection that induces the aggressive behavior in bees, for instance. And others make compounds which promote the gregarious behavior in locusts. So these are examples of behavioral changes, which are really very, very important for the survival or for the modification of the behavior of certain hosts. It's really quite remarkable. Well, a great example is the infection of grasshoppers by fungi, which causes them to climb up a stalk, impale themselves with their jaws on the stalk, and produce a fungus which comes out of their head, actually, and which is a way of spreading spores from high up, much more effective than spreading spores on the forest floor. So there is a connection between the gut and the brain via bacteria, and this leads to changes in neurodevelopment. It might cause anxiety and depression, perhaps Parkinson's disease, and who knows what. But it certainly does cause diarrhea. That much is known. So there is a possibility for explaining a number of psychological diseases as being microbial.
And if I could just add, Ilio, I think the problem here again is we have inference that maybe it's important, but we don't have good, solid evidence that it's important.
In part, because we don't have good animal models for some of this. We don't have a good animal model for anxiety, certainly for autism. Anyhow.
So if you look at those things that we talked about, I think one of the key things is that we've achieved a lot since 2007, but we have a lot of remaining basic science questions. And that basic science can lead to really important applications for humankind, right? So understanding microbial communities, we understand a lot more about them now, but there are questions about population genetics, how the different microbes in the microbiome interact with each other, about the physiology. them sharing metabolites, how that influences their survival, the stability of a population, the evolution of a population. Understanding these kinds of things will be really important for bioremediation, right? Now, when I talk about the microbiome, I'm not just talking about the human gut, I'm talking about the population that lives in the ocean. So when you think about the plastosphere, as Linda just talked about, understanding how those populations survive and how they function together is really critical to do that bioremediation. Climate change is a big thing. Microbes can be very effectively utilized for CO2 capture. Probiotics are something we've talked about for a long time, but they haven't lived up to their promise yet. Right? And instead of responding to an emerging infectious disease, how can we forecast where that emergent infectious disease is going to arise? Okay. Now, if you talk about studying microbes in culture and as single cells, we still don't fully understand the genotype-phenotype connection. So you get the DNA sequence and you can't exactly predict the phenotype from it. You can make guesses, but you can't know yet. We don't fully understand the extent of genome plasticity, how rearrangements, how substitutions influence what's happening in that organism. Physiology, structure-function relationships of a lot of the key components, secretion machinery, transcription machinery, translation machinery. There are still a lot of nitty-gritty questions about the structure and function. Molecular machines, how can we understand how those machines are working? Cell biology and synthetic biology also falls into this. And if you think of applications, there's clear biotechnology applications, producing molecules that benefit humans, agriculture applications, development of new biosensors that will be able to assay environmental health, improvements in drug delivery, improvements in antimicrobials. One of the groups this morning talked about antibiotic resistance. Improvements in antimicrobials so they can be more effective is one tool that we have to think about along those lines. Biofuels, and so on and so forth. A lot of really important basic science questions that are key to the applications. So a couple of examples of things that Ilio and I think are really important, right? Are actually that we're particularly interested in, I guess I'll say. So one is it possible to measure microbial physiology of populations? Can we understand the energy and metabolic flux that's going on between the different cells in those populations? Can we take a complex population and have a really good way of getting rid of one of the types of cells in that population, one kind of microbe, and see what happens when that microbe's gone? Can we do the same thing by knocking out a certain kind of gene in a microbial population? Why do bacteria have so many overlapping mechanisms to regulate gene expression? Right? It seems more complicated than it needs to be, which probably means we just don't understand it well enough. And a really important question for a lot of the question marks that Ilio posed with the behavioral issues in microbes that have to do with thinking about chronic diseases. How do you confirm causation in complex systems? There are a lot of papers that come out that say, this shows that X, Y, and Z is really important for autism. And when you read the paper, you say, I don't think it shows that, right? Maybe, but maybe not, right? But to answer all of these questions, and I re-emphasize this because it's so important, the pipeline of basic science is really key if we're going to generate those applications. And that is such an important concept because very often government agencies that provide the funding for funding agencies, that provide the resources for us to do work in our labs, don't understand this. And we have to always convince them.
que la microbiología es la reina de las ciencias. ¿Qué les parece?
Ciertamente, Elio, claro que sí.
Elio, you want to go from here?
This is the extension of the graph that we showed up 15 years ago. So after something of a dip at the time of the ascendancy of molecular biology in eukaryotic systems, genomics and metagenomics took over, and now the future is very bright for microbiological studies because so much is left. So here's the final thing. Les agradecemos muchísimo por su amabilidad de oírnos. Que les vaya bien. Many, many thanks.
Thank you very much. Gracias, Elio. I forgot to mention that Elio has a website, a blog, which is called, you have to remember this, Small Things Considered. Just put Small Things Considered in Google and you will find a great variety of topics that are discussed, very, very interesting topics. You will enjoy them very much. You can put Stanley Malloy's name and you can go through Facebook and other sites, and he has some outstanding lectures and that have to do with science education generally for the general public or even for scientists. You know, I remember Elio said one time that he likes questions that you cannot find the answer in Google. You can only ask to the religious scriptures. The Judaic religious scriptures have the answer. So as you can see, Stanley and Moselio live in this world where there's no boundaries between science and philosophy. I mean, most of the things they talked about are philosophical views of how we approach science, how we approach microbial problems. So with no further ado, we have a question by two of our students. Is Isela there? Isela Serrano? Or Christian Ortiz? Are you there? Are you in the auditorium? I think they're getting set up for that.
Ya aprendió, pero hola.
Okay, ya te escuchamos.
Hola.
So this is Isela, is one of our graduate students. And
Segura.
she has sí, se oye muy bien. She wants to ask you a question.
I'm really grateful for your talk, specifically when you say that the future is bright. So that's very powerful to me. And clearly, a lot of prophecies have been fulfilled since 2007. But maybe we can try to make another prediction, one that involves not only the techniques, but also the people that is doing the research. Well, let me elaborate. As you say, microbiology touches everything. And for example, the One Health approach, and due to the COVID pandemic, it has been become evident that the One Health approach is needed, but still lacks implementation at different levels, like geographic levels. And maybe this is due to the lack of funding and infrastructure or government policies. So the question is, how young microbiologists in countries with limited funding and resources can actively participate in the implementation of this concept beyond just publishing the research in journals, in order to make more accessible to a level region these concepts.
So I guess I will just start and say that one of the things that is a growing idea in science is that you really need broad groups of expertise to attack a problem. And it's called team science, and it's more complicated than it sounds, but it's this idea of bringing people together with different expertise and from different places who will look at the problem differently, but work as a team to solve the problem, right? Now, this was hard to do before we had a lot of the tools that we have for communicating from a distance. But I think now it's a very different thing. I talked to you a little bit about using GIS to try to identify a certain site. That is a project where I'm doing the GIS work in the United States. I have colleagues in South America and Africa who are setting up to do the drone work and the sequencing work. And all of this data will come together to try to give us an answer. And then the entire team will be on the publication. But we are distant places around the world and have different kinds of expertise. I might add one other thing, and that is that, microbial populations work because the diversity of microbes provides robustness to their survival in microenvironments. And the same thing is true with science. The diversity of people who are working on a problem are really important for making sure that you understand that problem well. And younger scientists are particularly valuable. Every one of us who's a senior scientist can say, very often, younger scientists in the lab bring a problem to us that we never would have thought of. So I think that these things will happen, and I think that that's part of this future.
Yeah. Thank you.
Thank you. So thank you, Isela. I'm going to read one question, and then Christian Ortiz, get ready. I'm going to read one question from Agustin Lopez Munguia, and he says, do you think that biotechnology, the applications of microbiology, are going faster than the advance in basic knowledge? I am thinking, for instance, on fecal transport.
I would let Elio comment on this, but he stepped out for a minute. But so I think that, let's just come back to the question that somebody asked Linda about, do you think you could use GMOs to clean up plastics in the ocean? And right now, fundamentally, I think we don't know enough. We don't know enough. We don't know if those GMOs will survive in the ocean. We don't know if they'll do the function that they do in the lab when they're in the ocean. And we also don't know if they might mutate to do something that we didn't want. So there are a lot of questions there. And I think that this is a big challenge with environmental biotechnology. A lot of hope, but it requires a lot of caution.
Stanley is one of the world experts on this question. So I'm lucky to have him, because if anybody knows the answer, he is it.
Great. So, Christian, do you have your question ready? I suppose Christian is there.
Yeah.
Go ahead, Christian. We can hear you. We can hear you very well.
Thank you.
Go ahead.
Talk. Okay. And the question is, in your paper, you described the first and the second golden age of microbiology. Try to describe and identify microorganisms and their metabolic and biochemical characteristics. Now, one of the principal applications of microbiology is the use of microorganisms to solve human problems. And the question is, what do you think about the ethical discussion between scientists and society about the application of engineering microorganisms into the environment?
Thank you. Go ahead.
I would like to ask you, what do you think?
What do you think, Christian?
I think that is a powerful tool. Is a I don't know.
Si quieres decirlo en español y yo lo traduzco.
Ok. Pues yo creo que es una herramienta poderosa. Creo que nos ha brindado y nos ha abierto posibilidades que creo que estaban casi que en la línea de la ciencia ficción. Y creo que bien administradas pueden ser una solución a los problemas que hemos muchas veces generado nosotros mismos, ¿no? La misma actividad humana.
Okay.
Entonces creo que...
Yeah, so he says that GMOs are a very powerful tool. He believes that they provide answers that you could only think about through science fiction. And so that he's in favor of using GMOs in the environment, but carefully regulated and carefully thought about.
So, Christian, I think that that's a very thoughtful answer. I think that the one thing that people are concerned about here is that very often in the past, we've done these big ecological experiments, and then things didn't work as well as we thought because we didn't understand them well enough. So, for example, rabbits were introduced into Australia because they thought that they would be a real effective food source. The rabbit population didn't have natural predators, and it just caused total destruction in Australia. Now, who would have known that, right? It seemed like a pretty simple thing to do. And there are so many other examples of that where we do something in ecology and you don't know what's going to happen. To me, this comes back to the other question, is that it's really important to understand the biotechnology as well as possible and to do the experiment on a small scale before you try to scale up to a bigger environment. What do you think, Elio?
Elio?
What can I do? I'm listening to the master. Who knows?
Can you repeat the answer, Stanley? Because the connection was not so good.
So I think that you have to be cautious. And so it's really important to understand the biotechnology as thoroughly as possible and to do the experiment on sequentially larger scales before you do it on a really big scale.
Okay, good. Now, I remember that was a fun story that pertained, Elio, about the number of bacteria in human cells. There's been discussion here at our institute about that. Can you comment on that? If Elio wants to do that, if he approaches more the microscope, the microphone, and then you can elaborate more, Stanley, on that story, please.
Surprisingly, we don't have a good handle about the total number of cells in the human body, so we can approximate it. And it turns out that there are probably more bacteria than there are cells in the human body. That's the answer. By how many? Maybe a factor of 10, but we're not quite sure.
But Elio, I think that the story is the initial projection we made on small things considered that took over everywhere and then was written up to be wrong in the Atlantic. anyway, Ilya and I made this prediction. It was based upon a lot of things that seemed like good science when we made this prediction. And it ends up that we were off by at least an order of magnitude. And it was very funny because this public science writer who writes very often for Atlantic magazine was the one who started exploring this together with some colleagues in Israel. And they did a really nice job of showing that we came to some conclusion without adequate data.
So I remember your calculation was 10 bacterial cells for every one human cell. And then it turns out to be more 1-to-1, right?
Yes, that's right.
Yeah, we've heard, we've read all about that, and yeah, so that's a funny story. So I just want to ask one question and probably leave space for one or two. I'm going to suppose I'm a graduate student. I'm going to about to graduate. I'm going to go on a postdoc. In which area should I go into next year? I mean, if you had to do that, what would be your advice? Which area of microbiology?
The environmental microbiology, microbial ecology.
Okay. How about you, Stanley?
I'm glad I don't have to make the decision now because there's so many exciting things that could be done.
Okay, all right. So anybody else has a question? ¿Alguien más tiene alguna pregunta? Let me see here in the chat.
I want to thank everybody for listening so carefully and for asking such intelligent questions. Ha sido un gran placer.
And I just want to say also thank you, and that you're going to have this slide deck. There's some references that are on the slides, but there's also a bunch of other references that are buried in the comments. So if you're interested in some particular topic, make sure to look for the comments. And it's not a thorough list of references, but it's something that will get you started. So anyway, muchas gracias.
There's one, I have a burning question. That was you in 2006 talked about developmental biology being influenced by microbiology. And I can think of different answers, but what's your feeling today?
So I, in the comments, there are some references to this that people might look up. One of the ones that set us off to thinking that in 2006 was some work by Laura Hooper and Jeff Gordon that showed that if you look at germ-free mice, the vascularization of the intestine was very low compared to mice that have the regular microflora. But if you take the germ-free mice and early on you add back the microflora, they develop a full normal vascularization, right? So vascularization of the intestine is a real important part of developmental biology. In humans, if it does not fully happen in the first 1,000 days, those kids are destined to have malnutrition for the rest of their lives. So that's a developmental biology question, issue that depends on microbes and has a real profound impact on humanity.
Great. Well, on that high note, thank you so much, Elio and Stanley. I congratulate everybody for this symposium. I'm very glad that our community got to meet you and know you better, and I'm sure they will follow your writings and your podcasts and so on and so forth. And we'll be attentive for the next publication of the Golden Phoenix. And thank you. Thank you very much. Gracias a todos. Adiós y gracias. Gracias a todos por su participación.
Okay, I think that I have to start, or there is any other introduction, or
Yeah, Dr. Nagabi?
yes, this is Mohsen.
This is Victor Mohsen. Can you start to share us your presentation, please?
Yes. Okay.
And then I'm going to start with introduce you.
Yes, you can start anything that you want.
Aquí. Bueno, vamos a continuar con la siguiente presentación. Es para mí un verdadero honor presentar al doctor Nagabi. Él es profesor de Ciencias Métricas de la Salud en el Instituto para Métricas de Salud y Evaluación de la Universidad de Washington. Él dirige el trabajo de este instituto sobre diversas causas de muerte, incluyendo las ocasionadas por desastres naturales, la guerra, terrorismo y enfermedades infecciosas. Ligado a esto último, por la resistencia a los antibióticos, que es de lo que nos hablará hoy. Para esto trabaja con colaboradores y gobiernos de muchos países. Solo para darles a conocer algunas de las muchas cosas impresionantes de su trayectoria, de su currículum, el doctor Nagabi ha publicado 633 artículos, cuenta con más de 300,000 citas a sus artículos y tiene un índice de H de 160. En los años 2015 y 2016 fue nombrado por la empresa de información Thomson Reuters, una de las mentes científicas de mayor influencia en el mundo. El seminario que nos presentará hoy se llama Métodos de estudio de la resistencia a antibióticos y resultados globales, regionales y a nivel de nuestro país. Creo que al menos parte de lo que nos va a presentar fue publicado este año en la revista Lancet. Dr. Nagabi, thank you so much to be here with us in this event. Please go ahead with the talk.
Okay. Thank you so much. Thank you for your introduction and thank you for the invitation. I am very happy to present some detail about the antimicrobial resistant or just for the bacterial pathogen that we did. And I will be happy to answer any question that you have. It is not a problem that if you interrupt me and ask your question or your comment, I will be very happy to answer. And also in the end, I will be happy to have any type of discussion with your colleagues in any part of the world. Okay. We started calculation of the burden of disease from 1994, and then in the 2010 we started new version of the global burden of disease that I think that every year or every couple of years published in the Lancet. And the last one is GBD 2019 that we published in Lancet, and new one that will be GBD 2021 will publish soon by the end of this year. And we hope that we can publish in the November or August. Some part of, in the global burden of disease, we calculate burden. It means that deaths, prevalence, incidence, years of life lost, years of adjusted with disability lost. for the around 300 diseases by age, sex, and location. Location means country, but for 22 countries, that Mexico was one of them, we calculate by the state and we publish. And also you can access to this result in the internet because all of our product, all of our production is public good and is free of access, free for the access. From 3 years ago, we started a project to calculate burden of antimicrobial resistance or part of the burden of antimicrobial resistance in the each causes of the GBT. I will explain more detail. And fortunately, by the end of the 2021, we finished this project, and in the first month of the 2022, we published this in the Lancet. And now we are in the process to repeat this calculation for the antimicrobial resistant burden every year. And we are in the process to start for the GBD, for the burden of disease for 2021. Everything that I will discuss is about burden of disease and antimicrobial resistance in the burden of disease in the GBD 2019. Then you do not have any expectation for the discussion about effect of the COVID on the burden of disease. It will happen hopefully by the end of this year. We will publish for the 2021. Okay, then if you let me, I will go through the what we did. I hope that everything, my presentation and my voice works very well. Okay, this is a flow chart that what we did. We had 10 steps in this process. I will try to summarize some important steps that it will be useful for our discussion, and then I will go to the result. First step, okay, first step, we calculate burden. Here is DALYs for the 300 diseases. And now I show just for the 2025 disease, the highest top, that is cardiovascular, neoplas, maternal and neonatal, other NTD, and blah, blah. These are DALYs that we had in the 2019 for the top 20 causes. But how many part of these deaths or this disability was related to the antimicrobial resistance? For sure, for example, many people die due to the cancer. But in the process of the dying or in the process of the treatment, some pathogen will rise and some infectious produce, and some of these part of infectious are resistant to the antibiotic. This person died due to the cancer or died due to the antimicrobial resistance by the cancer. Then we want to extract the number of the deaths or DALYs or VLL due to the antimicrobial resistance in the whole of the disease. For example, lower respiratory. Many people get lower respiratory and go to the doctor and get treatment. But some of them go to the resistant and they die by the lower respiratory with the antimicrobial resistance. Then antimicrobial resistance is a intermediate cause happened in the different process of the disease. Okay, now we want to understand what is the number of the deaths associated or due to the antimicrobial resistance. Then we calculate in the second step number of deaths in the different disease due to with the sepsis, not due to the sepsis, because sepsis is not underlying cause. Sepsis is intermediate cause. Then, as you see here, for example, in 2019, we had around 14 million deaths due to the different causes with sepsis. As you see here, we had 7 million deaths due to the all infectious disease that they die with the sepsis. 1 million deaths due to the neonatal, maternal, and nutritional disease that they die by sepsis. 5 million deaths due to the non-communicable disease that they die by sepsis. Then sepsis, it will be our envelope for the calculation of the antimicrobial resistance. Then between sepsis, or let me come back to the previous, between different disease and sepsis, there is one bridge. For example, if I have a stroke and I go to the hospital, I do not get sepsis unless I have one other disease that can be UTI, can be meningitis, can be lower respiratory, can be skin infectious disease. Then automatically every people that goes to the hospital or does not go to the hospital get different disease by the one infectious syndrome, he or she moved to the sepsis. Then we have to calculate what is the bridge, what is the connection between different causes and sepsis. As you see here, we clarified 11 infectious syndrome that can be UTI, can be skin infectious, respiratory infectious, peritonitis, diarrhea, CNS infectious, cardiac infectious, and bloodstream infectious. And you can see for the all cause, this pattern is different by age. For example, in the under 5, the biggest one is lower respiratory. Sick people, sick kids with the getting lower respiratory move to sepsis and die. And then after that, BSI, and then diarrhea, and then other non-bacterial infections, same as viral and trophozoid and fungal or other. Then we calculated this by the country, by the age, by the sex, by the year. And then after that, now we have infectious syndrome. We started to, I will explain how we calculated and which data we used. Just I want to say the story and then I will come back. For the each infectious syndrome, we calculated what is the pathogen distribution by the age, sex, location. And then,
Marcel,
yes.
Do you have a presentation? Because we do not see it.
I have presentation and I am showing my presentation.
Can you share your screen?
Yes, I shared my screen. Let me again share. Sorry.
We do not see it.
We are not seeing anything.
I am super sorry. Can you see now my presentation?
Yes.
And now, 1 second.
We can see it now.
And
Yeah,
now,
that's fine.
Yes, we can see it.
That's fine.
in the presentation mode.
Yes, we see it
Yes.
in the presentation mode. Yes.
Okay, then let me come back. Rather, I am very sorry. I needed this comment sooner. Okay. Here is our flowchart for the calculation of these 10 steps. And let me come back. This is the number of the DALIs for the 2019 for the different diseases. And I showed here top 10, top 25. And then we calculated for each disease, if there is any point rise, because I cannot see you and then I don't know. Then we calculated for each disease, how many percent or how many deaths happen with the sepsis. And then we calculated what is the intermediate cause between different causes and sepsis. And then we calculated pathogen distribution. That is the detail that I explained. If I missed some part or you have any question about this part, you can keep or you can rise, then I will continue and I will stop in some area for any question. Okay, now we calculated by every age, sex, location, infectious syndrome, what is the pathogen distribution? What is disease? Staphylococcus aureus. We had 1 million deaths due to the Staphylococcus aureus. These 1 million deaths in the Staphylococcus aureus happened some part in lower respiratory, some part in bloodstream infections, some in the intraperitonitis and E. coli and Staphylococcus pneumonia and Klebsiella and others. Then we have this pattern. Now, we need, if this calculation that if somebody has a resistant, how many percent or what is the relative risk of deaths with the resistant or with the, for example, relative risk length, increasing of lengths of stay in the hospital. Then we calculated relative risk, and then by the end, we calculated what is the burden of the drug combo. This is very short story. I will come back step by step for how we calculate. I know that it was not complete. Okay, let's go for how we calculated sepsis from the global burden of disease. We used multiple causes of deaths and hospital data that has discharge situation. And you can see that we used many data that was accessible. Maybe there are lots of data that is not accessible, but we gathered all the data in the world that it was accessible around 130 million records from the multiple causes of deaths and from hospital with the discharge result. And then we calculated for the every record or every disease, how many percent, because it is multiple cause, how many percent they die with the sepsis. And then we run a mixed effect binomial logistic regression, and then by age, by sex. And then we calculated 13.7 million deaths with sepsis globally in 2019. 5.2 was with the non-communicable disease, and 3 million was in the under 5. Okay, then it is a very big. Then this is the pattern of sepsis. You can see pattern of the fraction of deaths with sepsis in the correct word. And you can see, for example, we had 1.4 million deaths due to the cirrhosis. These 1.4 million, 42% die by sepsis. That from these, 500,000 was died by peritonitis, and a small part with the bloodstream infections. And COPD, if you look, we had 3 million deaths due to the COPD, 17% die by sepsis, and these 70 deaths, 17% was 160,000 by the bloodstream, community source LRI, hospital source LRI, and odd. And same as this, we have this for the every disease, every location, every age, every sex. Then now we should go to the how we calculated pathogen distribution. We gathered around,
Professor Magavi?
Yes.
I'm sorry to interrupt, but you don't seem to have your slides in the presentation mode. You have to click at the bottom so that you can.
Okay, I am in the presentation mode, but let me change a little, maybe, sorry. Let me stop sharing.
Sorry.
No worry, I am sorry that it was not in the presentation mode. What about now? It is in the presentation mode?
Yes,
Sorry.
now it is. Now it is. Now it is.
Okay, sorry.
Thank you.
Again,
Thank you.
I am sorry for the inconvenient process. Okay, let me come back. Okay, in the presentation mode, we can see more detail, and then you can see percent of the deaths by sepsis in all causes in 2019, and fraction of the deaths with sepsis in the different causes. And then how we calculate pathogen distribution. Pathogen distribution, we calculated with the around 400 million test result. we gathered whole data from the, I will explain that how we gathered, but if you want to say, for example, Central Latin America, I can find that South Asia, oh, where is the Central? Anyway, in the Central Latin America, you can see we used 8 million record for the calculation of sepsis, 3 million record for the calculation of the case fatality, 11 million record for the pathogen distribution, and blah, blah, blah. Okay, then we gathered a big database. And how we calculated pathogen distribution, because many microbiology research is not in the whole, doesn't have complete denominator. For example, you publish many papers that what is the main four pathogen for the lower respiratory in Mexico? What is the top 10 pathogen in bloodstream infectious in Guadalajara? Okay, that is very good. But in the classic statistic method, we cannot use these because we do not have complete denominator. What was the other? You do not report usually in your paper. Then we used a new method for the calculation with the name network meta-regression with covariate, that we pair two pathogens together and calculate based on that pathogen distribution for each infectious syndrome. And you can see here, much better. This is the pathogen, this is the number of the deaths due to the each pathogen in the each infectious syndrome. You can see that Staph aureus is the biggest killer, much more than malaria, more than HIV/AIDS. Nobody didn't look to that. And E. coli and Streptococcus pneumoniae, if this is number of deaths, if we move by the number of viral air, E. coli, it will be first because it kill in the younger age. Staph aureus kill in the older age, usually. Then we have this pathogen distribution. Now we have to come back. Now we have pathogen distribution, infectious syndrome, GBT cause, sepsis. What is the pattern of the resistance? Again, we gathered big database for the prevalence of resistance. And we calculated for the 88 drug combo antibiotic pathogen, what is the number of, what is the prevalence of resistance by location, not by age, not by sex, by location. And then, because there are multiple resistance, we made a method with the resistant profile that we use, some statistical method that we combine different resistance. And then, this is one example. What is the distribution of the Lrse? This is input data that we use. You can see we have not bad. It is good data. Some country like Kazakhstan, Mongolia, Tajikistan, Uzbekistan, and some country in the sub-Saharan Africa like Somalia, Yemen, okay, we do not have data. Then we estimated based on our method, based on the regional pattern, what is the fraction of the resistant for methicillin for the Staph aureus. And we did this process for the 88 pathogen. This is E. coli and cephalosporin, and third generation of cephalosporin. And you can see, again, we have almost enough data. For some of them, we have enough data. For some of them, no, it's not something that we can say for everything we had, same as this pattern. But anyway, it was based on the everything that was in the book. Maybe we lost some publication in Russian, in Arabic, we searched in the Spanish, maybe we lost something in the Portuguese, but anyway, this is the process that we are doing every year, and we will try to fix our problem. And this is another fluoroquinolone for the E. coli that you can see. This is the distribution of the resistant. Highest is in India, South Asia, Pakistan, Afghanistan, Bangladesh, and Iran, Iraq, and something in the Central Africa. And in your neighborhood, and also you are also in Mexico, it is a little high for the E. coli and fluoroquinolone that you know better than me. Okay. Now we had number of deaths for the sepsis, infectious syndrome by pathogen when we have prevalence of resistance. We are now trying to calculate relative risk of deaths with the resistant. And then we gather a big database. Our relative risk is not by country, is global, because we didn't have enough data to calculate relative risk of deaths or relative risk of the longer stay in the hospital by 88 drug combo by location H6. But it's not something that we say, okay, in the next year, we will try to calculate by the H6 location or region. Maybe we can find data and we can do that. Then now we calculated two types of deaths or two types of burden. When somebody died by the stomach cancer, but in the process of chemotherapy, he or she had lower respiratory infections, but we cannot, and lower respiratory infections had resistant, it was Klebsiella and was one other antibiotic. We cannot say that lower respiratory resistant to the Klebsiella in the lower respiratory was causes of this or stomach cancer. We name these associated deaths. But for the other, this is counterfactual replacement for the no infectious, counterfactual for the replacement of the no resistant. But for the some part of these deaths, we can say 100% deaths happened due to the antimicrobial resistance. Then here you can see this is Mycobacterium tuberculosis. Around 90%, 95% are susceptible. This is Staph aureus. Around 20% is susceptible. This part is just associated, and this part is attributed. Then this is for the every infectious syndrome, every pathogen. When we are saying every pathogen, 23 pathogen. Why we selected 23 pathogen? we did not select 23 pathogen. Process selected. Our standard was in each infectious syndrome when we have lower than 15% other, we will stop adding pathogen. And we go with the calculation of the different pathogen in the 85% of the infectious syndrome. When other will be lower than 50%, we stop. Then we calculated 23 pathogen. And for these 23 pathogen, we calculated 88 drug-bug combo. And you can see here, this is a number of deaths associated and attributed in the world by the pathogen. You can see E. coli, Staphylococcus aureus, Klebsiella pneumonia, and different other pathogens. We have 6 pathogens are responsible for the more than around 1 million deaths. Okay, and here is by location, by super region. In reverse order, that many people thought that, okay, antimicrobial resistance is a disease for the rich country. No, antimicrobial resistance is not for the rich country. It's for the poor country and rich country. When we decompose them, you are seeing Africa is the highest. When we are decompose this process, we will see in poor country, infectious disease is very high. Then tendency to the going to the resistant also will be high. But in the rich country, infectious disease is low, then using different antibiotic in the different wrong guideline or wrong process increase antimicrobial resistant deaths. Then there is 2 pathways. One pathway, infectious disease is high. One pathway is the antibiotic usage and antimicrobial resistant going high. And here is for the global. You can see this is methicillin resistant for the Staph aureus global. And 6 other is more than 50,000 deaths. Methicillin resistant is more than 100,000 with the Staph aureus, drug-bug Staph aureus and methicillin is 120,000 deaths, and other 6 is more than 50,000 deaths per year for attributed, not for associated. Pattern with the associated is different. Now look to the Latin America. Look to the North America and Latin America, PAHO country. This is age standardized mortality rate attributed, this part. This is age standardized mortality associated, this part. And okay, Mexico is in the middle, but do not forget that lowest is Canada, US, Colombia, Argentina, Venezuela, and some other. But is not specific. When we go by the drug-bug combo, we will find that which drug-bug combo is higher. And look here, maybe this was better. This is Mexico. This is deaths, age-specific mortality deaths by the different drug-bug combo. You can see by the male and female. This is red one is attributed, green one is associated. Now compare, for example, with Guatemala. Compare with, for example, Barbados. Compare with the, some other, Bolivia. And you can compare with US. We can compare with Canada. Where is Canada? Cuba or Canada. This is Canada. And then it shows that where do you have problem? But let me move forward. So what is the socio-demographic index? We made the socio-demographic index that it is a geometric mean between GDP, education level in over 15, and fertility under 25. This is socio-demographic index. And when we combine mortality due to the attributed to the antimicrobial resistance, we will see there is a very nice pattern. When you have higher socio-demographic index in this region, Haiti, Bolivia, Guyana, Guatemala will be higher, Mexico will be here, Canada, US is here, Cuba, I forgot. Colombia will be here, and this is the pattern of combination of the Latin America with the socio-demographic index and attributed. For sure, with the associated, it will be different. This is pathogen distribution. We sorted this country based on the age standardized Daly's rate by the attributed deaths by the pathogen. This is Mexico. And you can see when you go here, this is, for example, you are much higher between E. coli compared with the Staph aureus, because you have a young population. And Klebsiella is much more than of the Pseudomonas aerogenes. And you can see this is Mexico. You can compare with Brazil. You can compare with Colombia, with any other country. And if you look to the US Staph aureus is much higher than E. coli. And E. coli is much higher than others. Acinetobacter in US is very high. But when you compare ranking in Mexico, Acinetobacter is lower rank. But here is not, because Acinetobacter usually happen due to the secondary infections in the hospital. And this is the difference. And here is Daly's rate with the associated, that again, you will see that here E. coli is first, second is Staph aureus, third is other, and you can compare with US. We will publish this paper very soon, and we will share with all of our collaborators in the country very soon. Then you can put your comment on that. And this is age standardized mortality rate. Again, But when you go to the age standardized mortality rate, pattern is a little different. And okay, thank you. This was the whole thing that I wanted to say. And I hope that this is very summary. And we hope that we publish this paper for we submitted for Europe in Lancet. It is under review, and we will submit very soon for the PAHO, that what is the pattern of the antimicrobial resistant burden in PAHO. And I will be happy to answer any question, any comment.
Yes, yes, I have a, let's see. You can stop by sharing if you wish, so that we can, okay, we can see each other. Oh, thank you very much for your very thorough description of your studies. My question goes, I understand well that
I lost your voice. Sorry. I lost
the incidence of different pathogens related to respiratory disease.
El mundo, no se te escuchó bien, creo que se cortó la pregunta. ¿La puedes repetir?
¿Ya se oye mejor?
Yes, we are here.
¿Puedes cerrar la cámara?
Sí, sí.
Apaga tu cámara para ver si se estabiliza. Bueno, por lo pronto, ¿alguien tiene alguna pregunta? ¿De aquí?
Hi,
I'm
doctor.
sorry. I'm sorry. So you have this very complex pattern of incidence of various microbes related to antimicrobial resistance. My question is, can you see? Can you have general thoughts or general conclusions? Is the appearance of a certain pathogen associated to the use of a certain excessive use of a certain antimicrobial, or is the prevalence of a certain pathogen due to climate? Does it have to do with social and economic status? Do you have any generalities on the prevalence of pathogens?
Yes.
In different human groups?
Yes, we have this by country that it is important. Distribution of the pathogen by country that we will publish, and we will have in our VIS tool that will be public very soon. We have also something with the name of the DDD, drop day dosage or something. We have, for the different country, correlation between DDD and the prevalence of resistance for the different antibiotics. And also, we do not have anything now about the climate change, because it is very soon for us to go to this type of discussion. We will have, we have time trend for that from 1990 until 2020. And we will have forecasting soon for what will happen if we continue this way that now we are doing, or change the scenario that we are doing. This is something that I can say. For sure, we do not have everything.
Okay, thank you.
Thank you.
I think there's some more questions.
Okay.
Interesting to see the difference in Latin America. Can you comment on, please, a little bit more about the Mycobacterium tuberculosis, the high rate of mortality, regardless the antimicrobial resistance of this bacteria?
Now I do not have in my mind the distribution of the antimicrobial resistance for the TB, but we have in our public view that if you want, I can bring and I can show you what is the distribution of that by the world, and it is. I don't think that there is a big difference between Latin America with North America. It is the big difference is in the Africa and Central Asia and South Asia. They have a higher resistance for the antimicrobial resistance against of the TB. I hope that I answered your question, but there is, sorry.
Yes, thank you. And the difference among the different countries is related to the indiscriminated use of antibiotics only, or the socioeconomical condition of the countries?
I do not know. I do not know, because we just describe the situation. We do not, and we did not go to the causality. This is one other discussion. But for the very general, we know that in the poor country, infectious disease is much higher. And then in this country, due to the very bad situation in the hygiene in the hospital, selling antibiotic in the market, level of the resistance is high. But in the developed country, there is many other sources like inequality in the country. For example, here in US, we have big inequality. And then some part of this resistance is due to that, and some part is due to the wrong guideline and wrong using of guideline in the hospital. But there is one question in the chat that I prefer to answer also this one. Our suggestion is not just making new antibiotic. New antibiotic is useful, but this is not the main problem or main instrument for the fixing of this problem. Main instrument is making vaccine. If we make vaccine, our experience for example, for Haemophilus it shows very well in pneumococcus, it shows very well that making vaccine is much more useful than making just antibiotics. Then we are really push the world for the improving vaccination, making new vaccine, improving hygiene in the hospital, improving quality of the care, and making new antibiotic. I hope that I answered this question in the chat. It is from Ramses. Any other question?
Any other question?
Yes, I have one question. In some countries, some antibiotics are added to the food to feed the animals.
Oh, okay.
Do you have a kind of measurement of this correlation between?
We are working on that. We are working very hard to find correlation between using antibiotic in the animal, especially in the cattle, in the cow and pig and chicken. We have some progress. We had some progress. We showed that, for example, for some drug combo, there is a very good correlation, but anyway, correlation is not the causality, but it shows that there is a good correlation. For some of them, no, we couldn't find, and then we didn't decide to publish because we are in the first step. But it is a very, very important area that we want to work on that. Thank you. That was a very good question.
I have two questions, two general questions. How do you think about this estimation that 10 million deaths will be produced by resistant bacteria by 2050? I mean, is it possible, or how close we are?
Okay, we showed that now we have 5 million in the 2019 and 13 million in the sepsis. The paper that you mentioned, it is very important paper, and we answer that this paper is not complete because they included malaria, they included other things. This 10 million is not just antimicrobial, antibacterial resistant, and we passed this border from the many years. If we want to calculate that with the 2050, we have 10 million. Now we have more than 6-7 million deaths just for the bacteria.
The other question is, are the governments taking the required actions to control this problem?
No.
What do you think?
Now we share this result with the G7 Minister of Health personally. We are sharing this with the G20 in the November if it happens. We are sharing the result in the very short format for every government. Very soon we will send this result. And also we will put this in the public available that any government or any country can use, any university, any scientist, microbiologist can use that.
There is another question in Zoom. Can you see?
Yes. I am saying that when we do not have infections, we do not have antimicrobial resistance. Then we have to decrease infections. If we cannot decrease infections, then we have to fight against the antimicrobial resistance. That is my point. That's why if we want to make a priority, which one is the top priority? For sure, prevention with vaccine is the top priority. And if we cannot make vaccine, if we cannot have a good coverage, then fighting against the resistance is the second step. I didn't say that we have to select vaccine or fighting in the resistance. I hope that I answered to this. The example is now we can see the example that now it's happened for pneumococcus. The example is for Haemophilus. The example is for the meningococcal meningitis, that we have a very big decline. Okay, this is vaccine. there was not antibiotic. Then we prefer to make this comment that it will be good for the vaccination. Sorry, I stopped if there is any question.
Well, thank you so much, Dr. Nagabi,
You must welcome.
for your great talk and at the same time, very worrying. And thank you so much.
Thank you. Thank you for your invitation. And I hope that you can access to our result very well and use our result in the government, in your university and with your student. And all of the time, we will be happy to receive your comment. We are sure that we have some mistake. We are sure. If you send our mistake, your comment, we will be very happy because we can fix that in the next version. Then please contact us with any comment or any problem that you find in our process that we published. Thank you so much.
Thank you very much.
Thank you.
Les recordamos que a las 4 es la siguiente plática. No falten, por favor.
Sí.
Muy buenas tardes a todos. Con esta plática, el día de hoy terminaremos nuestra sesión, nuestro microcongreso, que pues se preparó con mucho cariño para celebrar los 40 años de nuestro instituto. El día de hoy tengo el placer de presentar al doctor Robert Edwards, que bueno, él es el actual director de Bioinformática y Microbiología Humana del Acelerador para la Exploración del Microbioma Humano FAME, por sus siglas en inglés, en la Universidad de Flinders, Australia. Sus principales contribuciones han sido pues alrededor de la microbiología justamente, de entrada, estando interesado en la regulación de la fijación de nitrógeno, y posteriormente en la regulación de la virulencia en salmonela. Justamente con esta regulación de la virulencia, él empezó a incursionar en la bioinformática, en donde pues se afianzó de manera importante y después empezó a trabajar con el Argonne National Labs y a través de una fellowship for the Interpretation of Genomes, FIG, en donde desarrolló el sistema RAST y MGRAST para la anotación de genomas y metagenomas bacterianos. El doctor Edwards es miembro de la Academia Estadounidense de Microbiología y fue nominado al Calvin for Teams Fellow por la Academia Nacional de Ciencias de los Estados Unidos. Ha publicado 100 artículos aproximadamente, tal vez más, y ha enseñado tanto microbiología como bioinformática por más de 20 años, en donde su labor en las cuestiones de docencia ha sido ampliamente reconocida de manera internacional. Ha ganado más de $10 millones en donativos para llevar a cabo su investigación. And for me, today is a really, really great pleasure to introduce Dr. Rob Edwards. He's also a really, really nice friend for many of us here in the Institute, and especially for people in our apartment. So very welcome, Rob.
Gracias, Rosie. Y buenas tardes a todos. I'm very excited to be here to help you celebrate 40 years of IBT at UNAM. And thank you very much for the invitation to talk today. Let me share my screen. Today, I'm going to tell you some of the work that we've been doing to find phages and viruses with machine learning. When I talked to Rosie initially, I came up with a title about machine learning and microbes, and she said it was a very boring title. So I tried to come up with a more exciting title, but I failed. So in the last 30 years or so, we've been developing software tools for phage and microbial genomes and metagenomes. And as Rosie said, when I was at Argonne, I developed MGRAST, I developed RAST, Patrick, the seed, lots of different tools. And so I thought I would tell you a little bit, un poquito, about some of the new tools that we've developed, and then how we're using those to analyze some of our data. I'm going to tell you several stories today, and then we'll see how many that we get through. We may not get through all of them, but let's start by taking a look at viral metagenomics. So the problem with identifying viruses, as we all have experienced with COVID, is that culturing is really difficult to do, and identifying new viruses is very, very hard. But they're so important in causing disease. COVID has wrecked the world, and we found it by DNA sequencing. So we go and look at different places to identify viruses. And we really think about three different situations. In the first environment, we have a very host-dominated place. This may be a sample from a human, from a coral reef, something like that, where there's a lot of host DNA involved. We may have an environment like maybe a fecal sample where we have a lot of bacteria, but there's still some small proportion of host DNA that we need to identify. And then we may also have an environment that is mostly dominated by microbes with a little bit of host, if any. And we need to think about each of those three environments differently. And so, for example, we may be looking at a tissue biopsy or a sputum sample from a cystic fibrosis patient. And there we have a lot of human DNA that we need to remove. As I mentioned, we may have a stool sample or something from our skin where we have a little bit of DNA. And then in the environment, it's mostly microbial and just a little bit of virus. Nowadays, when we do metagenomics, we start with viruses and we do a multiple step enrichment. And I'll tell you a little bit of details about each of these steps as I go through. Ultimately, we end up with total nucleic acid. But we don't start with this nucleic acid. What we actually start with is a mixture of double-stranded DNA, of single-stranded DNA, of double-stranded RNA, and single-stranded RNA. For the RNA viruses, we'll do a cDNA synthesis. For the single-stranded viruses, we'll do a second-strand synthesis, and then we'll sequence them using either Illumina or MinION. And so we've developed this new pipeline to analyze viral sequences from all of these different kinds of environments. In the lab, our extractions look like this. We start with an homogenized sample. We just remove the bacteria, the lumps, the solid parts. We might do some filtering to remove bacteria or remove sharks or remove corals, depending on where the sample comes from. We might concentrate the bacteria. We'll kill the bacteria with lysozyme, and then we'll do an automatic DNA extraction. But we were really curious about if we're losing viruses or if we're able to follow the viruses through all of these steps. And so we've been using flow cytometry to understand what happens to the viruses as we go through those steps of enrichment. If we take a sample, this is a sample where we've homogenized everything together, and we've got viruses, we've got bacteria, we've got some human samples. We get a lot of particles. And in flow cytometry, we'll measure the size along the x-axis here, and this is usually forward scatter or side scatter, and then the fluorescence, so the amount of DNA. And we've labeled the DNA here with cyber green so that we can detect it. And so we started with a very simple question. Do we lose the viruses when we do these filtration steps? And so we compared before filtering, after filtering with a 0.45 micron filter, and after filtering with a 0.22 micron filter. And you can see that we keep most of the viruses when we have a 0.45 micron filter. We also included a step where we concentrate the viruses from about 10 milliliters down to a single milliliter for further analysis. And we use these Centricon filters where you put the filtrate here and it passes through to the bottom. And again, that we can see before filtering, we've got our viruses and our background noise. And then after concentrating, all of the viruses are captured in the concentrate, and hardly any viruses flow through into the filtrate. So we're very excited that this protocol allows us to enrich just for the viruses. And then the next steps are either to sequence using the MinION or to sequence using the Illumina. This is Maria, who did a lot of our MinION sequencing in San Diego. And yeah. So once we've made a metagenome, how do we find the viruses? The solution that we have is to develop better software to identify virus sequences away from everything else. And we've developed a software pipeline we call Hecatomb. And this is a collaboration between my group and a group in St. Louis in the USA. Our approach is to create a pipeline that's easy for everyone to install, that's automatic to get the databases, and that makes all of the processing much more straightforward. We tried to balance having a big database that requires lots and lots and lots of computers with just a smaller database that requires less computing resources, but allows you to dig down into your data. So we don't need terabytes and terabytes of storage, and we don't need massive supercomputers to make this work. We've tried to make it easy to use, and I invite you to use Hecatomb. It's described here. And if anybody has any problems or any questions, please email me. I would be very happy to answer any questions. The outputs from Hecatomb include these graphs that show the alignment length. And so this is the length in either nucleotides or amino acids of our sequence similarities, and the percent identity. And this is a sample from a human, a stool sample from a human, where we've been looking at the viruses. And you can see each dot here represents an individual DNA sequence read. And you can see the kinds of viruses that we're retrieving from this stool sample. We've colored them so that we know whether they're RNA or DNA viruses. And what's important is that when we zoom in, for example, on one graph, we get 4 different areas that describe our data. In this corner, we see shorter reads that have less similarity. And these are likely to be things that aren't real. These are likely to be false positives. In this top corner, we see longer matches, shorter matches, but with very high similarity. And these may be things like maybe repetitive elements or transposons or short mobile sequences that are indicating also a false positive match. When we find longer reads, but with low identity, that suggests that we've got something new and exciting. These viruses down here are perhaps not exactly the Picorno by RNA viridae, but they're a relative of that. We've got long similarity, but not very close similarity. And then in this top right corner, we have most of the reads that are actually the virus that we're looking at. And so you can see that for each of the viruses that we say is present in this sample, we have a mixture of these 4 quadrants. These are probably not real. These are certainly real. And so now we can start to break this down and interrogate this data to ask more details about what is happening in each of these similarities. And so to show you that a little bit more, I'm going to show you a different data set. I'm going to show you a data set where we tried to identify a virus that's involved in a deadly disease. This disease is called nodding syndrome, and it's endemic to this part of the Congo and South Sudan. And
this is a giant village in Omoro County, Gulu District, where we came face to face with reality, away from government positions and public relations statements miles away in the capital, Kampala. 14-year-old Grace Achiro looks in bewilderment at the world, betrayal on her face as she probably ponders on her ordeal as she faces the day-to-day torments of the nodding syndrome. In a minute, she's standing, and in the next, she's down, succumbing to the violent seizures that come with the nodding disease. A short distance away is her younger brother, Walter Achora, who has as well fallen into the nodding trap and goes through the same cycle of torment.
So this is a deadly disease that affects young adults in Uganda and southern Sudan. There's no known cause. It may be caused by Onchocerca volvus, which is the cause of river blindness, but river blindness tends to be in the elder population. And the treatment for Onchocerca volvus is ivermectin, and ivermectin does not relieve any of the symptoms associated with nodding syndrome. So we got some samples from these patients, and we have blood, we have buffy coat, which is when you spin blood down, it's above the red blood cells, between the red blood cells and the white blood cells. And then we've got the cerebral spinal fluid as well. And then from the healthy children, we just have blood, which is all we could get ethically from these patients. And so we passed our sequences through Hecatomb. And this is Cara Henry Cox that has developed some of the pipeline that we used. And we could show that there was no known virus that was associated with disease. Every time we see a virus that's present in our patients, we also see the same viruses present in the healthy controls. And so we tried to use machine learning to identify the cause of nodding syndrome in these patients. We took the DNA sequences and we separated them out using a random forest classifier. And we had the classifier say which ones look like nodding syndrome and which ones look like controls. And when we test those sequences, indeed, these are the ones that come from the patients, and these are the ones that come from the control samples. So we can query each of these sequences from the random forest and say, tell us which sequence is most important in separating the control and the patients. And so, for example, this is the most informative sequence. and these are the next informative sequences. And so we can identify the sequences, the DNA sequences, even though we don't know what they do. And we can say, tell us which ones separate out the healthy patients and the control patients. Here I'm showing those top 3 sequences and the abundance of each sequence in plasma, in Buffy code, in CSF, and in our healthy controls. And you can see that for each of the 3 sequences, there's a big shift where we have lots of sequence in the patients and not very many sequences in the healthy samples. And so now we're trying to understand what those sequences are, how are they contributing to nodding syndrome, and how are they causing a disease? How are they involved? We think that they're viral sequences, and we're trying to get some more patient samples to test for the presence of those sequences in viral samples. So I told you a story about using Hecatome to look for viruses in humans, but I'm a microbiologist. So let's go back to looking at viruses in bacteria. And so I want to tell you a story about viruses that infect bacteria and archaea, and of course that's the phages. We've known about phages for many years. Phage lambda was isolated 70 years ago and has been extremely well characterized. And as you all know, viruses infect bacteria, replicate inside the bacteria, and kill their hosts. Now, I don't need to tell everybody at UNAM that there's been a resurgence in the interest in phages. Everybody's excited about phages for their use in phage therapy to treat especially these bacteria that are becoming more and more resistant to antibiotics. And we've also been involved in some work using phages to protect the warfighter from some potential bioterrorism threats as well. So there's a lot of interest in using phages. And that's because we normally think that phages kill their bacterial host. But that's not always the case. Many times the phage infects the bacteria and enters the lytic cycle and makes new phages, and bam, kills the bacteria. But sometimes the phage enters the bacteria, integrates into the genome, and replicates as part of the bacterial genome. And so we were interested in identifying these phages from within bacterial genomes. So the most common approach is to take a bacterial genome and to do something like BLAST to identify the phages that are present in the bacteria, and then to use sequence similarity to identify those phages. But I asked one of my students to identify phages in bacterial genomes without using sequence similarity. And so we came up with several different approaches to do that. The first is we looked at the transcription orientation of genes. If you look in a bacterial genome, as you undoubtedly know, the average bacterial operon is about 4 to 5 genes. But in phages, the average operon is about 30 genes. And so, for example, here's crassphage that we identified a few years ago. The blue genes are all on one strand, and the orange genes are all on the other strand. And this is quite normal for phage orientation. We also looked at AT and GC skews. And basically, if you have some flanking DNA in the bacterial genome, you can compare the AT and GC content with the region that you think is a phage, and very often, but not always, but very often, the phage has reduced AT and GC composition. And then if you take pairs of genes that have similar functions, phage genes are always smaller. And so here we have bacterial genes shown in gray, and we have phage genes shown in black. And the phage genes are about 2/3 the length of the bacterial genes. And I think that's because there's a pressure on phages to reduce the length of the proteins so that they fit inside the phage capsid. So we developed a machine learning tool to identify prophages in bacterial genomes using those features that I told you, plus a few other features. And then we've used that tool to characterize where the prophages lie in the bacterial genome. So we decided to compare how our predictions worked with all of the other predictions that people have done. And there have been lots of tools that have been developed over the years to identify prophages in bacterial genomes. Some of them have been more regularly updated than others. But we took all of these tools and we put together a platform where we have a set of bacterial genomes where we know the answer, and we can then compare the predictions with the actual the known answer. We've done a lot of work on looking at sensitivity and specificity. All of the tools have areas where they do very well and areas where they don't do quite so well. But what I just wanted to emphasize is that in this graph on the left, some of the tools take a very long time to run. This is in time in seconds, and some tools run very quickly. And in the graph on the right, some tools take a lot of memory and other tools don't take much memory. And the reason that I'm emphasizing that will become apparent. Because we wanted to know how many prophages are there. In GenBank now, there are 1.2 million bacterial genomes. We've used our program called PhySpy to predict the prophages in almost 1 million genomes. And the remaining genomes either are very, very short fragments where none of the contigs are very long at all, or they're not annotated, or there are other issues with those genomes in GenBank. From that, we found almost 40 million regions that could be prophages, but then we filtered them. So many of those regions are very, very short, less than 5 kilobases, and unlikely to be an active prophage. And then we asked for regions that have at least one match to a protein, a phage protein in the database. And from that, we retrieved about 5 million prophages. So way more phages than anybody's ever seen before. So we're interested in where these prophages come from, what's driving them, how they're distributed across taxa, where they insert, and how can we use this information to learn more about phage biology. So we started with 5 million prophages, and we combined that with all of the metadata around the genomes to start to answer the question about what's driving the presence of prophages. And today I'm just going to tell you a couple of short stories. I'm going to tell you about the genome length, the taxonomy, and the countries of origin. So when we look at bacterial genome length, so on the x-axis here, we have the genome length in base pairs. And we have on the y-axis, we have the number of prophages that we find. As the genome gets longer, we find more prophages. That's the green line. But actually, if we normalize the number of prophages for the total genome length, that's the red line here, once we get above about 2 megabases, we always have about the same number of prophages per megabase of bacterial genome. So what that means is that regardless of the genome, the bacterial genome length, there's a carrying capacity. The bacterial genome can only hold so much prophages per megabase of DNA. Once you add more, the cost of replicating that additional DNA outweighs any potential benefit from having that prophage around. When we look at the median genome size for all genomes, the median prophage size is about 35 kilobases. But when we look on individual taxa, we often see this unusual distribution of phage genome sizes. And something that we don't understand at the moment is that each taxa has its own profile of bacterial genome size, of phage genome size. So here's Salmonella, here's Strep pneumo in light blue, here's C. jejuni, Listerias, and each different taxa has its own distribution of sizes. We're not quite sure what that's telling us. I think it's related to the virulence factors associated with those phages and the sizes of those bacterial genomes. When we look at individual bacterial taxa, what we find is that most of the phages are from the bacteria that we've studied for many, many years. So the proteobacteria, the Firmicutes, the Bacteroides, we're really, really good at identifying phages in bacteria that we've studied for many years, especially bacteria from our guts. But we're missing millions of phages from all of the rare diversity of life that we've only recently started studying. And so my lesson, my take-home message is to understand more about phage biology and to understand the importance of phages. We need to start focusing on some of these rare members of the biosphere and perhaps move away from some of our usual suspects of gut bacteria. We also need to start, or need to continue, I should say, studying bacteria from different countries, especially Mexico. So this graph shows the number of different isolates from different countries. And of course, there are most bacterial isolates from the US, from China, from the UK. And we need to start understanding some of the rare biosphere that exists in the rest of the world, so that we can understand what that biology is providing to the great biosphere we're trying to understand. When we looked across time, we saw this beautiful graph that seems to show that newer bacterial isolates, and so the x-axis here is the year that the bacteria were isolated, and the y-axis is the number of prophages. It seems to show that the newer bacterial isolates have more phages, but that's not true. It's just that we have way more bacteria isolated in the last 10 or 20 years than we had previously. And so all of our observations are now skewed by the great biology that's been happening in the last 20 or so years. So I think these insights that we're learning from bacterial genomes are showing us how phages are evolving, where phages are coming from, and what they're doing to the bacteria. I'm going to skip over where phages insert. I'm going to tell you, hopefully very briefly, maybe not, come on. Maybe I won't tell you that. All right, I'll just tell you where prophages insert. We'll keep going. I was going to skip that, but I'll tell you it quickly. Most people think that prophages insert in the tRNA gene, but it's not true. When we look at insertion points, what we find is that most phages insert in defined insertion points that are not the tRNA. They're inserting between proteins. As I hope you remember from your basic biology, when a phage integrates into the genome, there's a recombination event between the bacteria and the phage, and that recombination event results in at L and at R. We can identify those sequences because they result in a duplication event where we have the same sequence in the bacterial genome. And within PhySpy, we've identified those core regions from all of the prophages that we've identified. And this is telling us something about, this is telling us something about phage biology. Lost there. There we go. This is telling us about phage biology. It's telling us the different kinds of enzymes the bacteria are using to integrate into the DNA. So in our core sites, we can identify mostly the lambda-like phages that are using a tyrosine recombinase. There are other kinds of phages that we have a much harder time identifying where they insert. All right, let me see if I can skip this time. Because I want to jump and tell you about I wanted to tell you in the last few minutes about how we're doing protein functional annotations. And this is work that has been done by Adrian Cantu that some of you may know. Adrian was a master's student in Cuernavaca and then did a PhD with me in San Diego and is now a postdoc at the University of Pennsylvania in Rick Bushman's lab. And so we've been interested in identifying, in trying to build better ways to identifying phage protein functions. And phages are quite simple. They basically just have DNA surrounded by proteins. And so we were really interested in seeing if we could come up with a better way of annotating the phages. And Adrian, being brave, decided to take on that challenge. So Adrian came up with 9 different classes of phage proteins. The capsid protein that's involved in making the head, the base plate protein down here that's involved in infecting the bacteria, the tail proteins that are involved in attaching to the bacteria, and the tail proteins include the fibers and then the shaft, the portal protein that's involved in putting the DNA inside the capsid, and then the head-tail joining protein that joins the capsid and the tail shaft together. And then, of course, we have one other class of proteins that is all proteins. So Adrian designed an artificial neural network that takes as input different features from the proteins. We only have a single hidden layer, and then as output tells you the likelihood that that protein has that function. So as inputs, we used either 2 amino acids, so each 2 amino acids going along the protein sequence. We used 3 amino acids going along the protein sequence. We used the molecular weight, the length, the isoelectric point, and we use the entropy. And Adrian has also tried some other things, including reduced amino acid alphabets, to try and simplify this as well. So Adrian built this artificial neural net, and like all neural nets, it has lots and lots of parameters, lots of dials that you can turn to try and get the correct answer. In this case, we have about 300,000 parameters that we can tune in our neural net. So Adrian took the protein sequences of lots of different phages, and he curated them to make sure that they're correct, and he just removed a lot of redundancy from the sequences. And so he ended up with a database of about 160,000 proteins across these 9 different of about 200,000 proteins in total across these 9 different classes. And Adrian found that he could get very good predictions. And so here we've got our accuracy of our predictions for the different classes of proteins across all different kinds of proteins. Some of them obviously are not so good, like this minor capsid, we're not very good at. And that's because we have very few minor capsid proteins. We only had nearly 700 proteins compared to with many thousand proteins in the other classes. And so the accuracy, as always with machine learning, is very dependent on the size of the training set. And you can also see that in the receiver operator characteristic curves that show we're really quite good at predicting them. But again, it depends on the size of the training set, depends on the accuracy. And then finally, the confusion matrix also shows that we're better at predicting some classes of proteins than we are at others. And in work that Adrian has continued to do, we've just released a paper into the archive this week describing how we can use this kind of analysis to validate predictions based on repeated sampling and subsampling of data through a confusion matrix. So the last story that I want to tell you in just the last couple of minutes is how we can use phages to improve our annotations in a different way than the way that Adrian has done it. And so this is work that's being done by my current PhD student, Susie Grigson, and is based on the same idea that phages are quite simple and that we should be able to come up with better annotations of their genomes. And so remember that all phage genomes are circular at some point. So when we find them in a bacterial genome, they're linearized, but phages go through a circular intermediate. And so we can take that circular intermediate and we can reorient it. So if we identify a phage, a prophage that's linear, but is in a slightly different orientation, we can just recircularize it by taking the piece at the end and moving it, taking the piece at the beginning and moving it to the end. So regardless of where the proteins lie, we can just reshuffle the genome and end up with everything in the same place. And so Susie has taken prophage genomes and she's recircularized them so that the red protein here is the integrase gene. And then she's colored the other genes by the functions that they're performing. And so if we just zoom in on this little bit at the top, you can see that we've grouped the phages. So here's the integrase, and then we have the base plate, the replication genes, and then the tail proteins, and the lysine genes. And in this way, we're refinding or rediscovering something that's been known in phage biology for a long time, which is the early genes, the middle genes, and the late genes. The early genes being the genes that are involved in integration and lysogeny, the middle genes that are involved in making the capsid and packaging the DNA, and the late genes that are the genes involved in lysing the host and escaping from the bacteria. We've also done this with other phages. For example, these are the mu-like phages that have a transposon and don't integrate using a recombination event. And we can still see the same thing where we have the early genes, the middle genes, and the late genes associated with integration, packaging, and lysing. So I told you a few stories today. I didn't have time to tell you all of the stories, of course. There never is enough time for that. I told you that we've been building some tools to identify viruses from metagenomes and phages from bacterial genomes. I've told you that most bacteria have phages and that there's lots of ways that those prophages are affecting their bacterial hosts. I didn't have time to tell you about how phages die, but the short version of that story is that phages are a trap for transposons. So I'm going to end because I wanted to leave some time in case there are any questions. And I'd like to thank you for your attention. And I'm going to apologize, as I always do when I present at IBT, for not presenting in Spanish. And I know that many, many years ago, I promised both Rosie and Jose Luis that one day I would present on Espanol. And that's still a dream, and I still haven't given up on that hope. It's lovely to see so many of my friends, and I wish that I was there with you to celebrate your birthday. And I'm going to celebrate your birthday later. I have a little Don Julio Setanta to enjoy in your honor this evening. So thank you very much, and I'd be happy to answer questions, and I can see Edmundo already has a question ready.
Very nice talk, and you went through it as in your usual self as a great teacher, and it was all very clear. And I suppose my question is, do you think that you have found all the possible phages that you could find in those bacterial genomes? I mean, are there any, I mean, everything was so rational, what you said, but do you have any wild guesses? You know, I mean, like something that could be so different?
I think that's exactly what we're trying to find. Yeah, definitely. I love that idea. And the question is, how do you prove that? So
sure.
we have lots of speculation about things that we think are phages, but we've got no evidence whatsoever that they're phages. And so now I've got some students that I'm trying to have them grow some of these strange bacteria that I've never grown before, you know, that grow in really different environments. And then to see, can we induce them? What happens if we throw the normal things like mitomycin or antibiotics, and then see, you know, can we use some of those old skills from the days when UNAM IBT was just starting? Can we use some of that old biology to identify those phages and show that they're really prophages? Yeah, absolutely. That's what we want to do.
Maybe some UV light.
Yeah, UV light, mitomycin, antibiotics, all kinds of DNA stressors. Exactly, exactly.
Well, well, as usual, it was a pleasure. And next time we have to do it in person with a few bottles. Several bottles. I know we can go through several bottles in one night. We'll just to tell Jose Luis and some of our other colleagues and friends, and I'm sure we can get rid of several bottles of tequila. Thanks, Rob.
I cannot wait to come back and see you all. It's been way too long.
That's correct. Thank you, Rob.
Thank you, Edmundo. Someone else here in the auditorium that has a question?
Your grandfather talking about.
Well, I want to follow the discussion that Edmundo started, because I was, well, not impressed, but worried every time that I see genomic data, that we really know, we have a really small proportion of the information that we can find or search into it. You show a graph, for example, in which you presented that we know a lot about phages that are in the organisms that we mostly studied. But for example, how about looking into beans? Because we have a lot of environmental samples that I think that they haven't been searched So, well, that's one of my interest, and I think that maybe you have think something about this.
Yeah, very much. We love binning metagenomes too, and it's a big problem. So for people that don't really think about this very much, you can sequence lots of metagenomes, and then you can use characteristics of those metagenomes to identify complete bacterial genomes. And this is metagenomic binning. It's very popular right now, and there's lots of papers where people are identifying hundreds of thousands of bacteria from different environments doing this approach. But the problem is, when you look at those characteristics, remember I told you that phages, prophages, are very different from their bacteria. They have different GC skews, different composition, different coding region sizes, and so on. And so those phages don't match with the rest of the samples in the bin. And so if you look in metagenome assembled genomes, they very often don't have any phages in them at all. And that's because the phages don't match with any characteristics, and the software that people have nowadays to identify the bins is unable to identify the prophages that correctly belong. And that's something that we're working on. And I have a postdoc that's developed some great tools that are not quite ready yet, but very soon I think will be ready for identifying prophages in bins.
Okay, thank you very much. Let me see if we have questions in YouTube also. Okay, I have one more. She says, Ana Sofia says, do you think there is a type of phage that could be generally more efficient in attacking bacteria with bioterrorist potential? Could such a feature be associated with genomic architecture?
Could you just say the first part of the question again, Rosie?
Okay, yes. Do you think there is a type of phage that could be generally more efficient in attacking bacteria with bioterrorism potential?
Yeah, absolutely. So of course, we think about lytic phages, not phages that are going to go temperately. But nowadays, what people are starting to do, and this was a big theme at the Viruses of Microbes meeting last week, is engineering phages to remove things that we don't want, to make them more efficient, to make them maybe broader host range, and to limit their ability to integrate into bacteria. And this is being used, it's been used successfully in phage therapy by Graham Hatfull, in cystic fibrosis patients, and it's also being used in bioterrorism as well. So can we genetically engineer phages to use them for our benefit, to make them stronger, to make them more efficient?
Okay, thank you very much.
Is there time for one burning question? One more?
No, we can have two questions. I am not looking at the Zoom, sorry, I was looking at YouTube. I have one here in the auditorium, and I don't see who is talking, but you can speak.
It says Mundo.
You again. Okay, yes, and then Lupita.
Okay, so in the end, Rob, what are phages for? What do they do? I mean, there are obvious things. I mean, they can transport genes, they can contribute to horizontal gene transfer, transposase, and everything like that. So the role is to leave and take genes from the bacterial chromosomes, or in the end, what's their biology?
Well, ultimately, the phage is there for itself. The phage, like everything else on this planet, the phage just needs to make baby phages to survive, right? That's what it wants to do. But many phages provide a benefit to their host. They protect the host against other phages. So there's a lot of evidence that many of the defense systems that bacteria have, like CRISPR-Cas, like restriction modification systems, like all of the new defense systems that people are discovering nowadays, that many of those actually were evolved by phages to prevent another phage coming in. If you're a phage hanging out in a bacteria, about the worst thing that can happen is if another phage comes and kills your nice, safe environment. But then, of course, you know, we all know about virulence genes that are carried by phages, toxins are carried by phages, and all of those things also benefit the host, because if they benefit the host, the phage makes more phages. So ultimately, it's all about making more babies.
All right, fair answer. Thank you.
That you find in that people, have you looked for them in other metagenomes or whatever?
You know, we're always looking through metagenomes to try and find phages and viruses, yes. And we're also always happy to help other people. So if you need help with data analysis, or if you want us to look for things in data sets you have, just reach out, and we're always happy to help with that, for sure. Yeah.
Thank you.
Okay, well, so thank you very much, Rod, for your nice talk. We are very happy to be closing this symposium, and we thank every speaker that we have today, and also you. And we are very, very, very happy to have all of you following our event. So, see you soon.